Hi, everybody. Welcome to the first and hopefully last NIH Freedom from Science lecture series. I'm Dr. Angela Rasmussen. I'm on the steering committee of the Save America movement. I'm also a virologist, and we have partnered with Stand Up for Science and 27 United to bring you this very special event today. I will be joined by several of my wonderful collaborators and co-authors, and we will be discussing the scientific evidence basis for the origins of the SARS coronavirus 2 or COVID-19 pandemic. The reason that we are doing this today is that NIH Director Jay Bhattacharya, and also currently acting CDC Director Jay Bhattacharya, is a champion of academic dissent and of intellectual disagreements being handled in a civil and scientific way. And as such, he has done no such thing when he's been discussing the origins of the pandemic. Unfortunately, Director Bhattacharya believes that the pandemic started as a lab leak that was funded by Tony Fauci through an R01 subaward given to investigators at the Wuhan Institute for Virology in Wuhan, China. He believes that the pandemic, the virus that caused the pandemic, SARS coronavirus 2, leaked out of this lab after virologists there were tinkering around with it. And that's what disrupted all of our lives extensively and horribly in 2020. The fact is, none of the actual evidence about the emergence of SARS coronavirus 2 into the human population suggests that any of that stuff happened. In fact, all of the evidence that we have to date suggests that just like SARS coronavirus 1 did 20 years before, SARS coronavirus 2 jumped into the human population through zoonotic spillover that resulted from the live animal trade at the Huanan seafood market in Wuhan, China, sometime probably in late November, early December of 2019. So my colleagues and I have done quite a lot of work to actually try to understand how the pandemic did emerge. We are all virologists, evolutionary virologists, pandemic experts, infectious disease specialists, and we really wanted to know how the pandemic started because that's how we would prevent another pandemic. Jay Bhattacharya disagrees with us, and so does the guest speaker that he invited for this event today. He's invited a gentleman named Matt Ridley, who is a British aristocrat who has a degree in pheasant mating from Oxford University. He's become a very popular science writer. He has written some very good books about genetics and things like that throughout his career. Unfortunately, he also has some pretty strong libertarian political beliefs, which have in fact colored the way that he responds to and analyzes scientific data. As a result of that, he believes that not only did the pandemic start because of a lab leak, but the only way to prevent future pandemics is to essentially stop doing virology research, or really any kind of research at all. And that is precisely why Director Bhattacharya has invited 5th Viscount Ridley to come and speak to the NIH, despite the fact that Ridley is not a US citizen. He does not pay US taxes. As far as I know, he's never been funded by the NIH to do any kind of scientific research, which he doesn't do because, again, his doctor in philosophy, his DPhil, is in pheasant mating. It has nothing to do with viruses. It has nothing to do with spillover. It has nothing to do with pandemics. And really, the purpose of today's talk is not to get an alternative scientific perspective on how the pandemic began. The purpose of today's talk is to justify what Director Bhattacharya is doing to the NIH by cutting significant components of our entire national biomedical research program. The idea is that if work in a lab causes pandemics, then we shouldn't work in labs anymore. And in fact, we shouldn't work in any labs, whether we're working with viruses or not, because there's all different kinds of dangerous risks that can occur with research. And we shouldn't be doing any of that. We should just be making America healthy again. We should just be drinking raw milk. We should just be treating our diabetes. We should just be going for a walk and exercising and, quote, eating real food. Unfortunately, those things are all very good for making you maybe more healthy, except for the drinking raw milk part. But having good overall general health does not protect you against specifically SARS-related coronaviruses or any other emerging virus that's capable of causing a pandemic. And what this is going to do is cause the entire budget of the NIH to be essentially illegally impounded by the executive branch of the government. We will have no research. We will have no ability to develop countermeasures. And we will be essentially sitting ducks waiting for the next natural pandemic to come along while we're so busy being worried about all of these invented scenarios that the director and his guest today have cooked up. So without further ado, my colleagues and I are very concerned about this. We decided that we would actually come in and correct them mid-talk to let them know that we are watching and we are actually going to hold them accountable to what the science says. So conveniently, I have written papers on this topic and I'm an expert on this topic. And I know a lot of other experts on this topic who have written these papers with me, including one of the leaders of this field who is here to discuss this with us today. And I'm very, very grateful that he was able to make it because his expertise is very complementary to mine. I study how hosts respond to new emerging viruses and how those viruses get into them and infect them and cause disease and spread. Christian studies how those viruses evolve and how they develop the ability to pop into humans and how they move around through different populations of hosts and what evolution does to them while they're doing it that can allow us to track them as they move from host to host, from species to species, potentially even in this case, causing a pandemic. So without further ado, I'd like to introduce my wonderful first guest, who is going to be fact-checking Director Bhattacharya with me, and that is Dr. Christian Anderson, who is the original lab leaker, actually, Christian, back in the day, and I'll let him tell more of the story, but he thought that the genome looked a little funny when it first came out, called up all of his friends and colleagues who are also experts in the field to say, I'm not sure that this is a natural virus. Maybe we should do a little more scientific research on it and see what we think then. And that's what they did. And they published one of, I think, the most important papers that has actually held up extremely well, considering how little data they had to work with when they wrote it, called The Proximal Origin of SARS Coronavirus 2, which came out 6 years ago on Tuesday. And boy, did the lab leak crowd have a fun time on the anniversary of the Proximal Origin paper, because they have been asking for it to be retracted because Christian changed his mind when he was presented with new evidence, which is actually the scientific method in action. But, you know, Director Bhattacharya, his guest today, and many of their colleagues consider it fraud. And so Christian has unfortunately been dealing with that for the past 6 years. But today he is here to talk about what we actually really like to talk about with this. And that is the scientific evidence that tells us that this happened because of zoonotic transmission.
Great. Thanks, Angie. And yeah, thanks for having me on. Yeah, as you mentioned, you know, trying to understand the origin of the pandemic is a pretty obvious question for many reasons. I think, you know, scientifically, because we're curious about that. But of course, as society, having dealt with COVID-19 for more than 6 years now, is that it's one of those key questions that we really want to know the answer to. And it's a simple question, right? We all lived through COVID. And the big question is, how did it start? And you need to know that, of course, because we want to try and learn from something like COVID-19 or prior flu pandemics or whatever you might have, to try and understand how we can mitigate that in future. I think trying to prevent it, I think is going to be very hard, but mitigation as these you know, things are happening, I think is critically important. So that's why we got interested in it, you know, early 2020, as soon as it was becoming clear that SARS-CoV-2 nCoV, as it was called at the time, is going to be a problem that we're going to have to deal with. And initially, when I looked at it, I've studied viral emergence, evolution for most of my professional career. So I knew how to tackle that kind of question. What I didn't know so much about, unlike you, for example, and other colleagues, is coronaviruses specifically. So when I initially looked at it, knowing what kind of work they were doing at the Wuhan Institute of Virology, for example, I got a little flummoxed for a while, I would say, where I started putting two and two and saying, like, this is Wuhan, this is where they have the lab, they're working on these viruses, they're trying to grow viruses, they're finding bats, they're also doing chimeric viruses. That looks fishy to me. And then there were a few features like a furin cleavage site and the receptor binding domain that, you know, on first look, just looked peculiar. And I put those together and immediately I thought, well, it's got to be the lab, right? And the question is then, it was like, okay, it's got to be the lab. Is it, well, there's many versions of how that could be possible. Is it just a bat virus they're trying to grow, or had they actually created the virus in the lab? Of course, these are not the same hypothesis. Very often these are put together and saying it's just one hypothesis. It isn't, right? All of these are mutually exclusive. But the first thing I thought is like, well, I got to talk to people about this, people that also know how to tackle these kinds of questions, people that are more senior than me and studied this for longer. Let's talk. And that's what happened in early, late January 2020 and early February 2020 also. And then, of course, that resulted first in the proximal origin paper, in which we presented our initial conclusions there. And then, of course, much more follow-up research since then, analyzing much larger data sets and actually taking a closer look at this. And yes, as you mentioned, it all points to like my initial hypotheses around a potential lab leak just do not hold up to scrutiny. And that's what we're presenting in the scientific papers.
Yeah. And, I mean, this to me is something that I've been thinking about for the last, well, since we started talking about it, which was sometime in 2020. I had been observing what was going on with the proximal origin paper on social media, and I had been dealing with some of the, politicians who were commenting on this and some of the political commentators and things like that. And it's just really striking to me, whenever we talk about this, we all talk about how little we know, right? Like I don't know anything about phylogenetics. And sometimes when we're having these larger discussions with our co-authors, you guys could very well be speaking Greek as far as I'm concerned,
Right.
Because I just don't know. You guys are making inside jokes about BEAST and all these tools and things, and I have no idea what you're talking about. But that is what science is all about. We all have our very specialized expertise, and we all can contribute something from that, and we all can learn something from each other. And that's one of the most rewarding parts of being involved in this work. It's one of the most interdisciplinary projects I've ever worked on.
Right.
But I think one thing that's really striking to me is that when you talk to somebody like Director Bhattacharya or Matt Ridley, they are experts in everything, according to them. They know how to clone viruses. They know all about viral evolution. They know all about spillover. They know all about wet markets. They know all about how the Chinese government works. They know all about how the Chinese mind works, which is actually another way of saying that they're racist. And they know, about everything. They don't need to ask anybody, and they never admit to not knowing something. And to me, that is one of the real markers of expertise. If you're really an expert in something, you know what you're an expert in, but you also know what you're not an expert in. And to me, that is one of the biggest differences in terms of the Zooinati, which is what the lab leak people call us, because we're like the Illuminati, except for, zoonosis and writing papers about it. But we all admit that we are good at, a couple things in this area, but it's a large field, and we need each other as a community to work through it scientifically, because it's a challenging problem. These other guys are so confident. They're like, well, somebody said this and we think they're credible, so it must be right. And that's actually a great segue to bring in our next guest, who is also going to be fact checking this with us, because as you know, Christian, I think 2023 was when it really started to come out. There was a lot of reporting that all of the various ICs, the different agencies within the intelligence community, had also been assessing the origins of the pandemic. And a couple of them decided, well, we think it might be kind of a lab leak, but they never shared any of the data that they based that assessment on. From reporting and kind of guessing and, talking to people who are in the IC, it seems that there's actually some disagreement between IC agencies about their different sort of lab leak scenarios. And as you pointed out, there's a number of different hypotheses.
Right.
People really kind of took this reporting with no data behind it. They did it in Germany as well, when it was reported that one of our German colleagues had met with the German intelligence community about the same thing. And they treat it like it's data, like it's fact. And it really is just a news report that, intelligence agency made a low confidence assessment of something. And so that's why I want to bring in our next guest, Gigi, because Gigi Gronval is a professor of environmental sciences, I think, at Johns Hopkins. Yeah. But really her specialty, it's very difficult to describe your specialty, actually, Gigi, because you're trained as an immunologist and you've worked in containment labs and you've actually done this type of research in the past. But since then, you've really been specialized in all different areas of biosecurity, biodefense, and a lot of this stuff within the intelligence community, the sort of behind the scenes government stuff that allows policies to be made and allows this research to be done, and also deals with the implications of this research. So what do you think, Gigi? I know you have a lot of thoughts on this about how, like, sort of those IC assessments have affected the public discourse on this. And in my opinion, I don't know what you think, but it's really driven the conversation away from a purely evidence and scientifically driven discussion, which in many ways it should be. Not saying that there shouldn't be an intelligence component or a forensic component, but ultimately at the end of the day, it's the scientific component that we rely on to tell us what happened. And I feel like a lot of this IC discussion is really a distraction from that, and an intentional one.
Yeah, well, first of all, thank you for the introduction. I really think that a lot of people think that there's like the man behind the curtain, you know, they think they have a lot of faith that somebody knows all. And that is really what a lot of this, you know, is about when you think about like what the, you know, this intelligence agency has had is changing their recommendation, like based on what, you know, what evidence are they using? And often they use opinions of experts that they pick. And so, you know, then it comes to like, well, who are those experts, you know? And so then you start to see like cracks in how they form their opinions. But this is an unfortunate part of this discussion, because a lot of the evidence on the intelligence side, of course, is not available for people to see. But I think people should be skeptical and think that, you know, if there was something that was extremely conclusive, that proved the point, that proved what the president really wants to be the case, we would all know about it. And so I think they should be really, you know, skeptical that there is some piece of evidence that they don't know that would show that it was a lab leak, because I'm pretty sure that would be immediately declassified, or, you know, would just be mentioned in a telephone call to whoever.
I mean, if there was that kind of smoking gun evidence, I find it hard to believe that it could be classified at any point. I mean, the evidence suggests that it wasn't a lab leak, so that evidence probably doesn't exist. But
I think that's a still common thing that people think in the lab leak world, that there's like that piece of evidence that, you know, that will prove them right. But, you know, we just don't know. We can't know what it is, you know.
Well,
no,
this
but
is getting.
I actually think like just one, because I do think if you look at the intelligence reports themselves, right, they do actually say that they don't have any evidence that the Wuhan Institute of Virology had, you know, either SARS-CoV-2 or a precursor that could have been SARS-CoV-2. I mean, quoting directly from the intelligence reports themselves, right, is that no indication that the VIV, so the Wuhan Institute of Virology pre-pandemic research holdings, included SARS-CoV-2 or close progenitor. That's straight from the intelligence community itself, right? They also say that information available to the IC indicates that the VIV first possessed SARS-CoV-2 in patients diagnosed with pneumonia of unknown causes in late December 2019. So this quite specifically, very specifically, in fact, says that of all the information, whatever that information is, because Gigi, as you're saying, we don't know what all the information is, but of all the information that they have, they don't actually have any information that shows that the Wuhan Institute of Virology had the virus prior to the pandemic. And in fact, the evidence they do had indicate that they didn't specifically. I think in terms of language, I think that's pretty specific, right? And I think if they had that information, that would say we have information suggesting that they did in fact have the virus, right?
I just want to clarify that I have received several briefings on COVID origins in a classified setting. So I and obviously you can't, you know, discuss what that is, but you know, I'm here. So I'm here.
Yeah, and I'll say I'm obviously quoting from again, President Biden asked for all this information to be made available. I'm quoting directly from the publicly available documents. But again, I would assume that if you have a summary intelligence report saying like, here's all the information, if there actually was any evidence to suggest that the Wuhan Institute of Virology had the virus prior to the pandemic, that would be mentioned in that summary document, right? And it is not.
I think another thing that people just don't, Angie just mentioned about how people in different fields don't understand what each other is talking about. But I think that goes, for other, not just in the scientific world, people who don't really understand the intelligence world or don't really understand how different laboratories could have different national laboratories could have different conclusions on things, and that seems more second nature to people who are like, oh, well, of course the FBI is thinking this versus the CIA, whereas to people who are not, who are not immersed in this might not, just understand that there's a difference or any distinction.
Yeah. And I will say too, that what it all comes down to is that, and we've had these discussions endlessly, they will be endless unless we actually see what they've based those assessments on. And this is, to get back to today's event, this is, I think, one of the things that Bhattacharya and Ridley and all of these folks count on. They count on there being enough uncertainty that you can say, well, the intelligence community thought this, and the FBI, CIA, like they're so important and it's classified and it's national security and all this stuff. It must be really, really good evidence. But as you pointed out, Christian, like they literally say we don't have any evidence, just kind of vibes and like we're suspicious. Like, it's not anything concrete. And they actually admit that. But because of the way it gets filtered through the media, and then because people like Bhattacharya, who are bad actors and who are doing this on purpose, will take that information and deliberately obfuscate it when they present it to the public. And that to me is probably one of the worst things that they've been doing. And it wasn't until, and we only have about 7 minutes until this theoretically starts. I don't know if it'll start on time. I'm watching it. So we'll just keep chatting until I see those two fine gentlemen, who are both quite tall, actually pop up on the screen. But, I think that one of the things that they've been doing is very actively trying to muddy the waters around what the evidence base actually shows. And Matt Ridley, the guest speaker today, whose talk is entitled Viral. Well, Viral is also the name of the book that Matt Ridley wrote with his co-author, Alina Chan, who also has a PhD, not in this field. She worked on, when she was a postdoc, she did 2 postdocs, and now I'm not sure what she's doing actually. But she decided to just sort of become a self-taught investigator, sleuth about lab origins, and figured that because she's a molecular biologist, she must know all about, she knows how to clone stuff. She must know how to clone a virus. And that's basically the technical premise behind this book that he wrote, which is more, they present it as we're lawyers arguing a case about which side is more plausible, which side has reasonable doubt about the origins of the pandemic. Was it natural? Was it a lab leak? And they say that they don't come to a conclusion, but the conclusion that they do come to is in fact lab leak. And the way that they do this is by not saying it was a lab leak. They say it could have been a lab leak that started the pandemic. And fast forward to an article that Jay Bhattacharya and Matthew Memoli, the principal deputy director of the NIH, wrote in a free market newsletter called City Journal, which is the official blog, I guess, they call it a journal, of a free market think tank, libertarian think tank. They wrote in this storied publication that the US's previous pandemic preparedness plan was send EcoHealth Alliance and Peter Daszak to Asia, give money to China, go virus hunting, bring a bunch of viruses back to the lab, juice them up with dangerous gain-of-function research, which for those of you watching who don't know what DGOF is, it is making a virus more transmissible or more pathogenic, causing more severe disease. So people are juicing up all these viruses that they just found. Then they're making vaccines to them because somehow, like, we're supposed to be making vaccines to new viruses and then stockpiling those. And then we're making a ton of money from the pharmaceutical industry. Right. It goes without saying that this is not what the U.S. pandemic preparedness playbook looked like. He's describing one part, a very exaggerated description of one part of an R01 project grant that was awarded to EcoHealth Alliance to do some virus discovery in China. It was not to juice up viruses. It was not to make vaccines for new viruses for a stockpile. It was to make us better prepared for pandemics, but this was a tiny fraction of it. Our pandemic preparedness plan had a whole lot more to do with making sure that hospitals stayed open and were not overwhelmed during a pandemic, because that's actually what kills a shitload of people in addition to the pathogen that is circulating. So this whole premise that virus hunting and reckless virology and a disregard for biosafety and, sort of trying to out DGOF each other because we're all competitive virologists who have no morals or ethics, is basically what Jay would like everybody to believe that the Biden administration did to prepare for pandemics, and before that, the Obama administration, and I guess the Trump first administration. But that obviously was not the pandemic preparedness playbook. And the reason that Jay is claiming that it was is because he wants to actually cancel all of this research. He does not want to fund this research because President Trump would like to have the congressionally appropriated NIH budget redirected to the executive branch so that he can consolidate his authoritarian control and continue the destruction of American democracy and American science. Now, that's just my personal opinion, but I think that that's actually a pretty accurate read of what's going on when we're talking about why people like Matt Ridley and why Jay Bhattacharya have continually, for the past 6 years, presented these conspiratorial alternative hypotheses for the origins of the pandemic as if they were fact, and now they are actually starting to make national policy based on them, which really should scare the hell out of everybody, because not only are we going to be completely unprepared for pandemics, we are going to be completely unprepared for everything. We're not going to have cancer drugs. We're not going to have new Alzheimer's drugs. We're not going to have anything. We're not going to have research, because the premise, again, is that all research is bad. So better give that money to President Trump, who will know how to better spend it. Probably on crypto or something corrupt, or I don't know, maybe another war that's going to ruin the world. There's lots of possibilities. Choose your own adventure. So, that's kind of, I think, where we're going with this. And what I think is going to happen, and the live stream has not started yet from what I can tell, but what I think is going to happen today at this lecture series is it's going to be just a lot of this trying to widen these uncertainty gaps to make this lab leak thing fit as if it were a fact, and then use that as a justification to basically can the entire American biomedical research enterprise.
Yeah, and I will say just one thing to that, Angie, because, as we're talking about pandemic preparedness, it's a lot, right? At the same time, it's not just biology research that is pandemic preparedness, but of course it's a key component. And I will say, during the first, Trump administration, during the first years of the pandemic, right, is that, the biggest success by far of the pandemic is Operation Warp Speed, right? The fact that we managed to get vaccines, effective and safe vaccines within a year is completely unheard of. In fact, people like myself was very skeptical when Dr. Fauci was talking about we're going to have this within a year. It was like, there's just no way. Things like this takes, at least 3 years. Yet there we were within a year, right, on President Trump's first watch. And I'll say like that's a big win to own fully. And of course was as a result of the key pandemic preparedness preparations that the United States have done over the years, right, which is basic research, basic funding into vaccine technologies, that then really came to the forefront as it was really desperately needed. And I think we need to remember that, right? Because it was under Donald Trump's first term, and I would love to see that kind of work continuing because it is so critical, right? We know there's going to be another pandemic. We don't know from which virus exactly. We don't know exactly where it will start, but we know it's going to happen. So be able to really be prepared to that will require the kind of programs that we saw really come to fore with Operation Warp Speed in the first part of the pandemic. Angie, now you're muted. Now we're running into technical issues. Yep,
can't hear you.
Can you hear me now?
Yes.
Yes. So you can't
Okay,
stream
we have
audio.
a slight problem. This is not being streamed over the internet. It is being streamed through Microsoft Teams. So let me get on there. The link that I thought was the video cast was actually a link to the Microsoft Teams thing. So hold tight one moment. And
that's so interesting that they're doing it that way.
You know what? It's probably the least effective way, and it's probably the way that Director Bhattacharya thought would be the most efficient, because that is his work process. So I'm going to take myself off the stage very briefly while I handle this so that you guys can't see me like ripping out my hair. You guys keep talking and I will be right back.
Well, just what you said, Christian. I mean, yes, it was a major accomplishment to have the vaccine. I also was so skeptical that we were going to have vaccine within a year. It just seemed fantastical to me. But it's not, from the very beginning, Donald Trump was undermining that effort. And so, any effort to that correctly cast COVID as the dangerous disease that it is, especially, for people who had no immune protection whatsoever. And especially for, he was constantly downplaying it. And he admitted to Bob Woodward that was his plan. He wanted to downplay it because he didn't want people to panic. And he especially didn't want the markets to panic. And so all of that downplaying it, not wearing a mask, not, questioning Tony Fauci's direction, like his advice, getting a cabinet member to write an op-ed that was against Tony Fauci. All of these things, there were so many things really added up so that when he wanted to claim credit for the vaccine, there were huge swaths of his supporters who didn't believe in the virus anymore. They thought COVID was a lie. And that's another psychological thing that I've been reading about, that some people have researched, that if people believe that a disease is not natural, they are less likely to take precautions against it. And this is something that was seen with HIV/AIDS. The people are much less likely to take any sort of medication or take PrEP or whatever it is if they don't think that it's a natural disease. And that's the case with COVID as well. So he was not thinking long term. I think that is a defining factor in his approach to governance. But if he was, then he would have if he believed that it could have had a vaccine in a year, he would have maybe acted differently. But he couldn't claim credit for it once it arrived.
Well, so I'll say, again, many mistakes were made during the pandemic, to be very clear. And I think, again, our response in the United States was very disappointing, right? Given how well prepared we should have been, and a lot of that was politicizing things that really should not have been politicized. So to be very clear, I completely agree with that. But I also think that we do have to take the wins that we got to take, right? And again, things like Operation Warp Speed required coordination across the administration and HHS to make sure that the money and funding was available to do the work, and then really pushing forward with the work. And I think that is a version of which, we should take that win, right? Because it was America that led the frontier, as we were talking about vaccines against COVID-19. Of course, my concern is that for all the reasons that you're mentioning and that Angie has mentioned as well, is that as we're looking to the next pandemic, I'm not actually sure that is going to be the case. Because our sort of pandemic preparedness has not improved as a result of the COVID-19 pandemic, right? It's severely degraded because we have moved away from what, again, should be sort of evidence-based approach to how do we deal with pandemics to something that is much more political and founded in strong beliefs that go one way or the other. And of course, the undermining of vaccine, confidence and the belief that vaccines are, in fact, as they are, and we know they are, safe and highly effective against a wide variety of different diseases, right, is extremely detrimental as we're talking about pandemic preparedness in general. And it's also not just, pandemics of future pathogens we have to worry about. It's also pandemics of endemic pathogens that were previously under control, like measles, for example, polio, as we keep looking further into the future. And that is to a large extent is because of the politicization of what, again, just should be fact-based, objective views of what works and what doesn't work. That to me is, and of course, a lot of that is what was done during the pandemic as well.
Yeah, I mean, just to mention that, was taking the win. There's a big effort right now. It's bipartisan in Congress to push back biotechnology, and that's great. But I don't think people understand how science works. That you can't just take the power breaker, turn everything off, and let it sit for a little bit, and then flip it back on. If the U.S. wants to do that after this really terrible disruption, it's going to take a lot of effort to be able to bring things back up to where it was.
Yeah. Yeah, and I think it's important, again, because not only it's not about power breaker, right? It's you burn down the forest and then have to regrow it afterwards, right? I mean, that burning it down does not take long. Regrowing a forest takes a very long time, right? These are certainly not the same. Angie, how are we doing on now you're muted. No, you just muted. So if you push the button.
There we go. There we go. It's too much muting, too many buttons. All right, I got it now. I got Teams working on my browser. So let's switch over there, and hopefully we will be able to hear the sound because it should be working.
Yeah, I'm looking forward to this.
Okay, I'm going to be quiet now.
In what follows, I have relied heavily on my co-author, Alina Chan, but if I get anything wrong in the next hour, it's my fault, not hers.
Oh, it's both of their
fault. I'm going to explain why I changed my mind, why I went from dismissing the lab leak hypothesis to concluding that it was not just probably, but almost certainly correct. This was not driven by motivated reasoning, quite the reverse. I am pro-science, pro-biotech, pro-genetic engineering. Indeed, that was one of the topics on which I spoke most often in the House of Lords.
I know he doesn't say he's
Although I am a professional writer, I do have some relevant expertise in this matter. I have a PhD in biology, I wrote a bestseller on genomics, and I was a member of the Science and Technology Select Committee of the House of Lords. But even if I had no expertise, as a human being who experienced the pandemic, I and everybody else has a right to investigate this question. This is not something where scientists should say, stay in your lane.
We've never said that.
And I am frankly dismayed by how the WHO, most of the scientific establishment, including some of the NIH and the science journals, plus most of the mainstream media and some of the intelligence community, not only failed to investigate this issue properly, but were reluctant even to discuss and debate it.
Correction.
If a plane crashes,
we did investigate it
we do our best to find out why and share the lessons. This thing killed 100,000 times as many as a typical plane crash. As you will hear, by far the most important discoveries about what happened were made by amateurs working in their own time and on their own dime.
Incorrect.
So it's worth, I think, starting with the question, why does it still matter 6 years on?
Because you're using
I think it matters for several reasons. Because we do owe it to those who died to find out why. We need to understand that last pandemic. We need to explain why the virus was unusually infectious for a virus that had just intruded into the human species for the first time. We need to prevent the next pandemic by learning lessons from the last one. And importantly, if we shrug our shoulders and say we may never know how this happened, that sends a message to bioterrorists that they can do this and get away with it. Attribution is deterrence.
Is he suggesting
not that I'm implying this was an incident of bioterror, but I think the next one possibly could be.
It's amazing that we have attribution technologies that get into like one stall in the market, and yet
viruses, the connection with Laos, the lab, the furin cleavage site, the diffuse proposal, and some loose ends.
Okay, everybody.
We're talking about a coronavirus. There are 4 kinds of coronavirus: alpha, beta, gamma, and delta. There are 4 coronaviruses that cause versions of the common cold and are endemic in human species, 2 alphas and 2 betas. There are 2 coronaviruses that have caused brief and lethal outbreaks in human beings in the current century, and there is one that has caused a pandemic. It is a close cousin of SARS. It is in the group known as SARS-like beta coronaviruses, or sarbecoviruses.
And it's actually called SARS.
What is the natural habitat of sarbecoviruses? It is the guts of one genus of bats, the horseshoe bats, which live only in the old world, not in the new world, are highly gregarious and tend to live in natural caves. So the question we have to ask ourselves is, how did a bat virus get into people? And ignoring possibilities like frozen food, which are not very plausible, I think there are 4 plausible routes. The first is that it was a repeat of SARS, in which an intermediate mammal host caught the virus and that infected people. The second is that the intermediate host was actually a person who went into a cave and got infected. The third is that the person was a scientist. And the fourth is that the scientist took the virus back to a lab and did research on it.
I'm confused about why 2 and 3 are different.
And the reason we need to take that last possibility seriously at the very start is because of the extraordinary coincidence of place and time. In all the many thousands of cities in China, and next to the 40,000 food markets in China, this outbreak happened in the very city where research on sarbecoviruses was occurring more than anywhere else in the world.
Also,
that is quite a coincidence.
It was occurring in at least 12
It's also a coincidence of time as well as place.
Every city
in all the thousands of years of human history,
coronaviruses.
This outbreak happened at the time when research was going on these kind of viruses for the first time. And lab leaks do happen and have happened. Just to run through some examples of recent and relevant lab leaks, there was a global pandemic of flu in 1977, which was derived from an inadequately attenuated
It'll be 50 years ago next year.
Virus used as a vaccine. There was an outbreak of anthrax in 1979 in the Soviet Union,
79
which
is not,
it took the Soviet authorities 12 years to admit was the result of a leak from a biowarfare lab. In Kunming in 2003, hantavirus infected researchers, and the person who investigated that case was Xi Zhengli of the Wuhan Institute of Virology. Same year, in both Taiwan and Singapore, researchers got infected with SARS while working on them, and again in Beijing the following year, at least twice, possibly 4 times, there was a leak of a virus. The important thing about those cases is that, except for the Taiwan case, they didn't know how it happened. So these things can happen without anyone knowing that there's been an accident in a lab. In Surrey, in England, in 2007, foot-and-mouth disease broke out on a farm just down the road from the world's reference lab for foot-and-mouth viruses. That turned out not to be a coincidence. A contractor had gone from one site to the other. And in 2019, just before the pandemic, brucellosis broke out and infected a large number of people in China as a result of a lab leak. And finally, of course, SARS-CoV-2 has infected people in the lab since the pandemic began, probably many times, but we know of one particularly clear case in Taiwan. All in all, in the first 20 years of this century, there have been hundreds of individuals infected in scores of labs and in many different incidents.
This is completely made up, okay?
And just before the pandemic,
in these specific situations,
the World Health Organization and organizations like the EcoHealth Alliance were warning that this lab leak risk was getting worse. The threat from lab-enhanced viruses is intensifying, and curiously, these warnings have been much quieter since the pandemic. That seems to me curious.
Is it curious?
Nonetheless, when the outbreak happened, the Chinese authorities immediately assumed they were dealing with a repeat of SARS, and that the Huanan seafood market in particular was where this was happening, and they went as far as saying that there was no human-to-human transmission, and they continued saying that up until the 20th of January, which turned out to be bad advice. in other words, the only way you could get this virus in January, they thought, was from another mammal. And at first, the evidence does look quite interesting, because the cases reported to the World Health Organization from December 2019 do cluster around the market.
I don't think anybody's ever gotten it from
the ones that had no connection with the market
or a marsupial,
than with the people who had actually been to the market. But we now know that that is largely a case of ascertainment bias.
Yeah, so this is untrue. I knew he was going to bring
this up. For whether or not you could even be considered for having this disease in December 2019 were that you had to either have been to the market or to live near the market. I'm going to briefly pause it for a moment,
so Chris, because I do feel that this part needs to be addressed. Go ahead.
Yeah, so what he's referring to is that between January 15 and January 18, it was true that part of the case classification was a connection to the Huanan seafood market for these 3 days. But of course, when we are looking at the early cases in December 2019, right, we are looking at retrospective investigation of earlier cases in which those ascertainments were in fact not in place. So we can bring up the ones we discuss afterwards. We can bring up, well, all of this is in the WHO report. So this is just outright false.
Bingo.
So I'm afraid this is purely circular reasoning, and it tells us nothing. Besides, we now know that there were at least 73 and possibly as many as 86 cases that were not reported to the World Health Organization from December 2019, and we don't know the origin of them.
Well, no
and indeed, according to a Chinese government document seen by the South China Morning Post, there were 9 cases in November. These have not been shared with the World Health Organization. They could be very important.
So this is really
furthermore,
important. This is again
the virus that people caught in the
where the WHO report comes in.
No, derived form, it was the B strain of the virus.
Sorry, Christian, go ahead.
Yeah, so to be clear, so he is talking about cases that were reported as potential cases in November of 2019 by the South China Morning Post and other outlets at the time. These were then investigated as part of the WHO investigations during the first mission to China, and are shown not to be COVID-19 cases. Again, flu at that time was highly prevalent, and these are likely flu cases. We know they're not COVID-19 cases. So that is just outright false.
Indeed.
And that's quite an important piece of influence, because it's very hard to see how that could imply an origin.
Sorry, Christian.
Sorry, there's some notes or something that came up on your screen now, so now we can't see his slides as well.
Yeah, this is what I get for trying to actually make this more visible.
An origin in the market.
How's that?
There were mammals for sale in the market and in other markets in Wuhan. Not very many by the standards of markets in southern China, where the sale of live wild mammals is much more common. But there were some, and they included animals that could be susceptible to this kind of virus. But it did not include bats, it did not include pangolins.
Which are irrelevant.
All of the raccoon dogs for sale, for example, in this market were sourced locally. They had not come from Yunnan, which is where
he doesn't know that, actually.
The people that got COVID were also sourced locally.
It is true that virus was present in the market, not just in people, but also on countertops, doorknobs, in the sewage, and so on. And those samples tended to cluster in the southwest corner of the market, which is where those mammals were on sale.
By the way, this is a paper that both Christian and I are authors on.
Until you find out that the Chinese CDC had prioritized those very areas for sampling. So again, this is almost certainly a case of
yeah, so he's talking about ascertainment bias again. So it's true that when we did the Warby et al., for example, we didn't know the underlying sampling density. We got that as part of the China CDC's own paper. And of course, in Chris Christoph et al. In Cell, where now we have the underlying sample intensity, whether positive or negative. So we can fully correct for that. So the idea that it's only positive there because that was sampled is, again, simply false, because we do have the underlying sample distribution and of course can correct. And the signal is that, in fact, there is a real clustering in that southwest corner of the market, which is not due to ascertainment bias. Again, we can discuss this more detailed afterwards.
Yeah, that sounds great. And I'll just add to that, a project that I have going on that I really need to get finished looks at host responses from some of these potential intermediate species at different places around the market. And at the other stalls that were not in the southwest corner where we do have animal samples, they're not the right species. And on top of that, even when we did have a species that was susceptible, we did not see any transcriptomic signatures that would be associated with infection with the coronavirus.
This is almost certainly a case of ascertainment bias of circular reasoning. And if you look for a correlation in those samples between mammal DNA and virus RNA, you find a negative correlation. In other words, there is a correlation between the virus and people's DNA, but there is not a correlation between the virus and any mammal DNA. There is one sample that has both raccoon dog DNA in it and virus RNA in it, but only one read out of 200.
Yeah, so that this again is incorrect. So again, in the clustering in the market that we see, we have one stall, for example, in which there are 5 samples that are positive for SARS-CoV-2, and that is the sort of epicenter of the clustering inside the market. And in those 5 samples, we see evidence of raccoon dogs and other susceptible animals in every single sample. So this is exactly what you would expect if you had infected animals that brought the virus in. You would expect to see them at the same time. But of course, you wouldn't necessarily, because they take many samples across the whole market, many of which are due to infected humans, is that this correlation between, it must be between infected animals that are susceptible and the virus, is a complete non-starter in the beginning.
Yeah, and I'm going to add something else to that has to do specifically with my expertise. So looking at these samples from the market, these are the worst samples I've ever had to work with in my entire life. They're supposedly RNA-seq samples. They are full of DNA. They are just a hot mess. And there is absolutely no way that you can associate the virus that was found in that sample with the animals that were found in that sample, because these are environmental swabs. They are not swabs that were collected directly from an animal. So they have a bunch of different RNA, lots of RNA from the different animals, as well as DNA, as well as viral RNA. The presence of any of those RNAs or DNAs in there is going to be based on where that swab was taken, when it was taken, when anything had been deposited on the surface that it was taken from. If there was any disinfection occurring, RNA degrades at different rates. Viral RNA degrades at different rates than host RNA because viral RNA is encapsulated, so it's slightly more protected. It's sensitive to temperature changes. We can never know most of these variables. So the bottom line is that a correlation analysis with these particular samples is a complete non-starter altogether, which is why our group never even attempted it.
Read out of 210 million reads in that sample maps to the virus. That is not strong evidence for mammals being the source.
What he's saying is just all about, like,
these were the reasons that led George Gao,
I'm telling you what the answer is. You know, trust me.
Quite early in the pandemic, that he thought the virus was not the source.
We've lost so much public trust. We have to trust these people in order to get it back.
It was this press announcement,
that's what they say,
rather obscure, but I did stumble upon it, that led me to begin to change my mind. I was gobsmacked by this. I thought it was case closed at that point. And I began to go back and look at the
so I guess that means he was anti-lab leak at first. He's sort of the anti-Christian.
I can't stress enough how different this is from SARS. In the case of SARS, infected civet cats, and in particular, people who handled such mammals, were very clear from the start. In this case, not only were no animal handlers infected of a relevant kind, but no mammal has ever been found.
Oh, I'm sorry, were there samples collected from animal handlers?
Not one.
No, and this is key, right? He talks about 80,000
reports that there were no animals.
The pandemic started,
80,000 sounds
did indeed start catching
80,000
from us.
99%
of
which
case
is
that precedes a human infection.
They're mostly like, you know.
So there's no direct evidence today that points to an intermediate animal origin
snakes and random stuff like that they found. I mean, this has
about
always
this
been a thing that they bring up, these 80,000
when
samples,
Xi
and they're just like
Jinping
random samples they got
published
from
the,
wherever.
Sequence of the
It's
virus in
convenient- it's
nature,
convenient
she
sample.
revealed something very important.
You
There
could
were
test
3
80 million, but if you're not
bat
testing the right
viruses that were
ones,
closely
you know.
related, one of them particularly so,
Oh, good times.
RATG13. And this is what she
Here
said
we
about
go.
it. We then found
Mojang mind
that a
time.
short region of the RNA-dependent RNA polymerase gene
Changing
from
the
a
name
bat
time.
coronavirus, which was previously detected in Rhinolophus affinis from Yunnan province,
But he's
showed
not
high
actually
sequence
trying
identity
to convince
to the new
anybody.
virus.
You know,
We
there's
carried out
not
full-length
like
sequencing on
this
this
is the way
and found it was
Bob Caddick does his
96.2%
presentations too.
the
It's
same.
just like, let me throw some information here.
There's
I'm not going to really
2
tell you what it
features of
means.
that passage
And
of
then
text
I have all
I need
these
to draw your
numbers
attention
and
to. First
letters
is they
and
clearly
graphs that are
imply
circled.
that they
It doesn't
sequenced the
mean anything.
virus
You know, he's
after
not
the
telling a
notice of
story. He's
similarity
just telling you a whole bunch of
between one
things
its
and then
genes
saying what it means.
and one of the human virus's genes. That turned out to be false. They had sequenced the bat virus in 2018.
They sequenced the
year
polymerase
before the
gene
pandemic.
of the bat virus.
In
I
which
should
case, why
add,
did
not
they
the
need to
entire
notice
genome.
just one of the genes being
Which
similar? Why
they
didn't they
published at
blast
the time, to be
the
clear,
Which
right?
they
had
published
to
at the
run this-
time.
whole gene?
And the reason that they look at
And
the
the other
polymerase
strange
gene
thing
is it's one
is
of the
that
most conserved genes throughout the
Notice the
genome.
sentence
So
about
if there's
the previous
differences there,
detection
they
of
can
this
see that
virus
this
does
one's a
not
little
have a
bit
reference.
different. But
Does
that
not
was
have a
about as
footnote,
much
does
as
not
they
have
knew.
an
It could have been like
endnote.
any other coronavirus.
I
in their
spent several
collection.
fruitless days trying to track down RATG13 on the internet. When had they found it? Where had they found it? Under what
In
circumstances?
2013, because that's what
It
the
did not
13 means,
exist. It was
guy.
a ghost. It took us more than 6 weeks to
And they found it in
resolve this
Tongguan
dilemma.
Cave, which is what the
And the work
TB
that
stands
did
for.
so was the results of Manali
RA is
Lahalka's
from
work
the-
in India
from
and
the
Rosanna
bat-
Rhinolophus
Sukker's in Norway.
affinis.
And what they
This
noticed was
year
that
Yeah.
there was a
they did that.
virus
So,
out there
yes,
that
it's all
had
there.
been described in 2016.
It's almost like we do
that had a
things
100%
systematically.
similarity to RATG13, but it had a different name. It was called 4991. And it later transpired that they.
Okay, so for everybody who
You
wants to
you
know
just
about
wanna
this,
also-
RATG13.
sometimes
at the
like we
start of
don't actually have our
2020,
naming system
but not
made
before,
up when we start taking
hadn't
samples or
communicated
start doing
this
an
fact.
experiment. So we
Now,
might
whether
use a different
intended or
numbering
not,
system. And
the
that's what
result of
he's
this
talking
was
about
to
here, as if
disguise
it's
the
some big
story
secret where
of the
they changed
closest
the name to
related
disguise
bat virus
this
and to
horrible
make it rather
polymerase
obscure
gene
at
that
a
they
time
found.
when it was quite
And
possible
you just want to pause for a
that the
second.
pandemic
Yeah.
would blow over as only
Because I think there is a key thing to know here, right? So initially, when they had the sample, this sequence, a fragment of it, right, and they give it the name of the sample itself. So it's a sample ID because they don't have a full virus. They
Right.
then later sequence the whole virus, or almost the whole virus, right? It's still just the sequence. They don't have the virus in the lab, but they have the sequence. But because they now have a full genome, that's why it gets renamed and gets a virus name, which is now RATG13. Right. So there's nothing mysterious. This is literally always done, right? And anybody doing sequencing of viruses would know this. So there's just absolutely nothing to it.
Yeah.
the pandemic would blow over as only a minor event in China. So what is the story of 4991 or RATG13?
Do
Well,
tell
the
us.
first thing is it doesn't come from Wuhan. It doesn't come from anywhere near Wuhan.
It comes from Yunnan
Southwest
province.
China, in Yunnan. It comes about as far away from Wuhan as the Florida Keys is from where I'm standing today.
Also, it's far away from
That's a
Guangzhou,
long
which is where
way
SARS-1
Right.
for a
started.
virus to travel.
Yeah.
And we know of only one animal that has made that journey from a bat cave
Now he's going to say humans.
to
Not
Wuhan.
true. The wildlife
It's
trade.
Homo scientificus. So what is the
Oh,
story?
for goodness
They
sake.
found that
Did we
virus
just
and
say Homo
under what
scientificus?
circumstances? Well, we did
That's a pretty
actually
good
have
one.
a report in the scientific literature from 2014 that was around the events that led up to the discovery of that virus. This article is actually about a different virus, a rodent virus, but nonetheless, it's about the same incident.
What?
But it wasn't until May of 2020 when a young Indian named the Seeker found a medical thesis from Kunming Hospital that gave us the
The
details
seeker, I
of
should
this
add,
story.
is like a
6
random
men
middle
had gone
school
into a
math
disused
teacher
copper
who
mine
sits
on a
the
mountainside in
computer all
Mojiang
day,
County
like,
in southern
doing
Yunnan
origins
to shovel
tracing.
bat guano out,
Hmm.
probably with a view to reopening the mine. They had got sick, badly sick with pneumonia. 3 of them died, 3 of them eventually recovered.
All of which is
And
published.
this was a case that
None
led-
of this is news.
All of it's public, and
Yeah.
I believe that they were hospitalized with a fungal infection.
that's the primary belief. Yeah, I mean, could it have been a coronavirus? Sure.
Of course.
Does it have any relevance to the origin of COVID-19? Right? Absolutely not. We're talking 2013. Clearly, RITG13 is not the precursor to SARS-CoV-2, right? So it is irrelevant, but it sounds bad.
Yeah, I'm going to take this opportunity to just actually point out something that we know, but I'm not sure all of our viewers do. And that is that, yes, RITG13 was the closest virus that we knew of in 2020 to SARS coronavirus 2. It was 96.1% similar, I think. That translates to more than 1,100 different nucleotide substitutions across the entire genome. So if they were to turn RATG13 into SARS-2, it's not only you just insert a furin cleavage site into the S1, S2 junction in the spike protein, you also would have to insert 1,100 mutations across the entire 30,000 nucleotide genome, which, you know, you could do it. It would probably be the worst, most tedious cloning project of your entire life. You could just get it synthesized, but it still wouldn't make that virus SARS-2. No, and no virologist in a million years would ever do an experiment like that, because we weren't trying to make viruses that we didn't know existed.
Yeah, I mean, that virus would fall apart, right? There's no way that would be a viable virus. Like, the idea that RITG13 could somehow have been the precursor is just off the table. It's not possible. It is one of those things that, like, there's many things that we know with, you know, high confidence, but we don't know for certain. What we do know for certain is that RITG13 is not the precursor of SARS-CoV-2.
Yeah.
I just want to just highlight something that Angie said, that is like throughout the genome. I think a lot of people think, well, if it was just like one chunk, you know, you could just like a paragraph in a Word document, but it's not that way. It's lots of mutations that it would, and it's just not how many years of evolution are encompassed in the difference between these two things. So it's not possible.
No, it's not.
Virologists all over China to get very interested. Samples from the men's lungs were sent to Wuhan and were probably the first and only occasion where human beings had caught a sarbecovirus directly from a bat.
What?
Which would be a very interesting story
in its own
right. And that led the Wuhan
EcoHealth Alliance put out a paper in 2022 that does suggest that people get sarbecoviruses directly from bats. The reason that we don't have sarbecovirus pandemics all the time is because probably most of the time when that happens, it's a dead end. Most of these viruses probably don't grow at all. They probably are not compatible with the human host because they just, you know, they can't bind the receptor. There's something missing in terms of something they need to interact with when they're replicating. For whatever reason, the viruses aren't very fit or may not even be able to infect a human host at all. So that's why we don't see SARS-related coronavirus pandemics nonstop. But people definitely get exposed to them through bats. I'm sure people doing exactly what he was talking about, going into mines that are full of bat guano, do get exposed to sarbecoviruses. But there's not that many sarbecoviruses. And this is another way that SARS coronavirus 2 is unique. It's not just that it's so infectious for humans. And by that, they just mean that its receptor can bind a lot of different ACE2 orthologs among mammals. It's a lot more than that. Like, SARS-2 is very unique because it actually is capable of replicating efficiently in a number of different mammalian hosts. And that's what we have actually seen since the pandemic has started. It's not just humans. It's adapted to grow in white-tailed deer. It's adapted to grow in cats. It's adapted to grow in dogs, because it's not adapted to grow in any one of them. It's a very effective generalist virus that can infect a lot of different species, much like many influenza viruses can, much like SARS-1 actually can. But it infects them to different degrees of fitness, and it also infects different individuals to different degrees of fitness. So that's why we have a lot of people that probably do get exposed to sarbecoviruses, but we don't see pandemics all the time, because also this probably happens out in the country in like a cave, where people aren't going to be in a densely populated urban environment and allow that pandemic to spread on when they do get a virus that actually can't replicate in that.
And Angie, we should talk about this more, because again, how does it set up from you know, an individual infection to a pandemic? But one key thing here too is that he has now concluded that they were infected with coronaviruses, right? And again, the official conclusion is that they're more likely got fungal infections.
Yeah.
So this idea that he just says, I think it's a coronavirus infection, so it's a coronavirus infection, where the official conclusions are actually was not a coronavirus infection, right?
And I should add, again, this man's scientific expertise is denying that climate change exists and pheasants mating.
in its own right. And that led the Wuhan Institute of Virology to mount a number of expeditions to the site, about 7 over the next 2 years, in order to sample the bats in the mine shaft. And you can see them here dressed in full PPE and handling live bats in a keep net. We'd like to know more about this site, but journalists and others who've tried to get there since the pandemic began have been turned away often very firmly.
I wonder why
there has been nothing published, literally nothing.
It's really shocking that we don't have good relations with China anymore.
No reports of whether or not
no, I feel like I should have the right to go poke around some abandoned mine if I feel like it.
And so these are some of the
I have more to say about that.
Need to know about 4991 or ITG13. Why did they change its name?
Because they had the full genome.
Why did they not cite their own 2016 paper in which they described the
They're quite busy, but they did mention that they had done it
Why
before.
did they mislead us about the date on which they'd sequenced its genome?
They probably
Why did they omit a connection with a human
They didn't.
disease?
They published the paper at the time.
And why did they claim wrongly, as it turned out, that there was no more sample after we finished the genome sequencing?
What?
Because there probably wasn't.
In the summer of 2020.
You know how much RNA is in those samples most of the time? Not very much.
Is a good word.
Oh, good.
2 sleuths,
the whole
one called Baba, one
in
called Francisco de Assis,
contribute their insights.
spotted in a genetic database that there were 8 other viruses that had apparently come from
Oh, was
the
that
same
RTG18 or what are they
and were
called?
closely related
Yeah, it's one of the other
RATG13.
Yeah.
We eventually this was
I love
eventually
how it's just so
confirmed to us.
deliberately soporific. You know, this is so damn boring.
Yes, we found 9 viruses altogether in our expeditions to that
They like
mine.
to say that I'm
Can you just
a
pause for a
terrible
second?
person,
Which
but
means
I wish
that
I'm interesting.
Yeah.
Go ahead, Christian.
Yeah. So just to be clear, so again, Xi Zhangli at the time, right, were doing scientific presentations showing these other viruses that they have identified. In fact, that's how these sleuths could find it, is because Xi Zhangli herself presented slides with that data showing that they had these additional viruses. What's really important, though, is that those viruses clearly completely unrelated to SARS-CoV-2, right? And somebody like me looking at phylogenetic trees all the time, it was very simple to say, like, yeah, well, of course they do, but it has nothing to do with SARS-CoV-2. And plus, if those were somehow the magical precursors or whatever, of course, Xi Zhangli wouldn't go around doing presentations on it. And again, they published a paper on it once they had finished the scientific studies, done the sequencing, done the analyses. A paper was published as soon as that was done. And indeed, they're unrelated to SARS-CoV-2.
Yep. Oh boy, it doesn't want to restart.
Not 8.
There we go.
Which means that the Wuhan Institute of Virology virus was detected in December 2019. The seeker eventually found a thesis the way he does.
Why are we giving this person the title like this?
They're described as lineage 4, being used MSc thesis, and which shows their relatedness and includes 4991.
I think I need to rethink my branding.
2019, which is how we know they haven't yet changed the name.
So these are not 9 close relatives, and they're not the closest relative to SARS-CoV-2, right? So again, this is just made up. But also really importantly is that these are publicly available theses. Like the sort of crime here is that they're in Chinese, right? I mean, that's hardly a crime, right? Given how many people speak that language, it just doesn't happen to be not me, right? So I can't easily access them. But what the seeker did was to get them translated so Westerners like myself could read them. And indeed, in there's no viruses closely related, or certainly not closest related, to SARS-CoV-2.
Well,
right?
just
so
wait. I mean,
if they were hoping to find that, it was actually the opposite, right? Because they might have hidden something that would have been SARS-CoV-2. And in fact, what we then find in these theses is very clearly not SARS-CoV-2.
Just wait a couple of years, you know, they won't bother to translate any of their research because the journals will all be in Chinese anyway. That we'll have to learn that language. So,
well, they won't be able to. They won't publish it. I mean, they won't publish it in a place where Westerners can read, or they won't care because we aren't.
They are the ones that are funding science.
Yeah, there won't be Western science because this is going to be the excuse actually to undo it all.
Thesis was published as a paper in Infectious Genetics and Evolution. Lineage 4 was omitted altogether. That may have been because they were saving it up for a later publication or something. We don't know. But
90% of their arguments are like
that they had lineage 4 in their possession at all.
That indicates absolutely nothing. If you
and again, lineage 4 is completely unrelated to science.
It's irrelevant.
It's totally different virus, and they have published that as a paper, right? I mean, there's no mystery here whatsoever.
Also, just as Americans, we really need to remember that having a British accent does not give you extra credibility.
Yeah, actually, that's a good point. I should have maybe brought this up in the very beginning, but I should add that when I mentioned that Matt Ridley is a British aristocrat, I mean that his family goes back to like, millennia ago. And his ancestors were not only supporters of King George during the Revolutionary War, but they were also supporters of the British during the War of 1812 because, they were British, which makes sense. But those two wars were basically trying to undo America. So I'm just saying that, he's maybe not the most trustworthy person, given that his family has been in the business of undermining America for literally like 15 generations.
I was just thinking more of like the dating game sort of comment, because like, how the British guy always won. So anyway, go on.
Yeah.
At all, or what they were doing with it. Then in 2021, there was a complication in this story because the Pasteur Institute found a virus in Laos that was more closely related to SARS-CoV-2 than RATG13.
Oh my God.
96.8% rather than 96.1 or 2%.
Imagine that.
Well, that seems to change the story. But it doesn't
There's a lot of viruses out there.
Yeah,
neither of these viruses is the ancestor of SARS-CoV-2.
Right out of the water.
They're not close enough to be the progenitor, but they are representatives of the group from which this virus is derived.
Okay, and so
pandemic preparedness. I really think that's the conclusion here.
Yeah, actually, pandemic preparedness research is really important because you know what? When we were looking at these viruses, when they were first discovered, actually, I wasn't looking at them at all because they're just another one of the many coronaviruses that have been discovered out there. We don't really know anything about them. Some of them are probably harmless. Some of them could be the next pandemic. How will we know if we don't do experiments? So I tend to not think, holy shit, like we just found another virus and it's even closer to this other virus than this other one that we found. Like, big deal. Like, it's another virus. There's tons of them. There's millions of them. Like,
that's why
we only care about the ones that are going to cause us a problem. He is showing nothing, as we pointed out repeatedly, that suggests that any of these viruses would be a problem or that they're involved in any of this in any way. We were doing this work to understand these viruses better as a group of viruses. We were not doing it to find like the next big pandemic that we could juice up and make even worse to cause a pandemic with.
Also, something to point out, he's pointing at papers that you guys wrote, that other people wrote. They're not pointing to any papers where he's had to logically lay this out himself that has been published in the scientific literature.
Because they haven't written any.
Because they haven't done that, yes.
The only paper that he's written has been either an opinion column in The Spectator, which is generally politically conservative, or it is his book, which he's still trying to sell. It's out in paperback now if you guys want to get it. It's one of the best selling-ish books that have been written on the pandemic, and I don't recommend it because it's full of untruths.
If the library has it,
all running a little behind his live recording here, so should we?
Yeah, we're gonna pick it back up.
And besides, the fact that they come from a different country doesn't necessarily change anything because the Wuhan Institute of Virology was receiving viruses from Laos via the EcoHealth Alliance, as shown by both emails and research reports.
I believe we're inactivated.
And indeed, genetic data.
What?
I believe those were inactivated samples and not actually live viruses, right? So that's having a sample does not equal having a virus.
So viruses were crossing international borders.
Yeah, and contrary to popular belief, as well as Matt Ridley and Jay Bhattacharya's belief, it is not trivial or easy to clone coronaviruses. So even if you have the full sequence, you can't just hit the reverse genetics easy button and make it.
She meant that they had been sampling bats in the vicinity of Wuhan in Hubei province extensively over preceding years and had never found these kind of sarbecoviruses there. So they had also been sampling human beings in Wuhan and had not found SARS-like antibodies in them. So no wonder she worried, could they have come from our lab?
Of course she did.
So I got to say something here. It's true that they didn't find any bats or any people in Wuhan, like when what he's just talking about. However, in 2003 and 2005, they did find farmed raccoon dogs and palm civets just outside of Wuhan in Enshi province, or Enshi prefecture, that did have antibodies and had fragments, I believe, of a spike from a SARS-related coronavirus. So we're not sure like where that virus came from. I'm not sure. Christian, you might know better than me, but definitely animals that were being farmed for sale in live markets, locally sourced animals, were infected with sarbecoviruses in Wuhan 20 years ago.
She eventually concluded that they had not, but we'll have to see whether that's plausible.
So she was honest, and she was worried about it, and she wanted to look into it, and she found that it didn't come from her lab.
No, of course.
So we're going to have to think
Peter
carefully
Daszak
about
of the
that.
Health Alliance in her laboratory. Peter Daszak was funneling millions of dollars from the US taxpayer to the Wuhan Institute of Virology to support her research. This is one of the NIH
their
grants.
Their sub-award for their R01 was, I think, around 100K, which is a tiny
but also to be clear, like collaborative research, right, is not funneling money, it's doing research together.
The next step was to identify other
that's an excellent point, Christian, because what they make it sound like is when we get a grant, it's making money rain on you. And the fact is that covers people's salaries and it covers the
Indeed,
stuff
Andrew Rambo on the 21st of November 2019, just as we now know the pandemic was starting, wrote on Twitter that the more we look, the more new viruses we find. The problem is we have no way of knowing which may be important or which may emerge. There is basically nothing we can do with that information to prevent or mitigate epidemics. So he's saying the benefit of this research may not be very great.
That's not actually true.
I don't think that's what Andrew said there.
Yes, we wrote a whole paper on this, and of course, this is one of the disagreements that Peter, myself, and our courses have had over the years. Right, is that this type of research is basic research, right, and is critically important so we can understand the viruses and that are out there. But in terms of direct pandemic prevention, right, we're saying we want to prevent the next pandemic, i.e., it doesn't happen. That's where I don't believe that type of research will do that, but will help mitigate and help us develop better therapeutics and vaccines. Of course it will, right? And again, we wrote a paper in 2018 in Nature on that exact case, which is what Andrew here is referring to. Andrew was a co-author on that together with me and Eddie Holmes. Yeah,
that's correct, Christian. And I agree with some parts of that. We should have another show sometime talking about this in particular, because we actually do really need to think about how we're distributing our resources for pandemic prevention and response and preparedness work. But you're absolutely right. And this is just emblematic of this entire conversation. I notice also that Matt Ridley did not mention that Andrew Rambo is one of our collaborators and our co-authors and has written extensively on this and is probably one of, I'd say, the world's greatest living evolutionary biologists who understands this stuff probably better than almost anybody else. And what Matt Ridley just did there was look at his fellow British person's very thoughtful, very insightful comment about something that's incredibly complex. And he reduced it to, well, he doesn't like any of this kind of research either.
Right.
SARS-related coronaviruses identifying more than 50 novel strains, sequencing spike protein genes, identifying the ones that bind to human cells, using recombinant viruses and humanized mice to see SARS-like signs. So live infectious experiments.
Transgenic mice that express the receptor for the virus and nothing else from a human.
Yeah, and to be clear, like, because recombinant viruses can mean many things, right? And again, we know this, like, the Wuhan Institute of Virology, for example, have VIP1 backbone, which they use as part of these recombinant viruses, which allow them to test different spikes of different coronaviruses, right? Again, SARS-CoV-2 is very clearly not that. Right. But really importantly, too, is that you can do pseudotype viruses, which are done very often, which are not actually replication competent. They're not infectious viruses, but you can use that to test, can they bind to this spike or can they bind to that spike? And I'm sure that's exactly what Peter is talking about. He turns this into, like, they'll create these Franken viruses, right? And they can infect all sorts of things. And that's just not correct, right? It's a misrepresentation of what basic biological research really is.
Yeah. And, I think about this a lot, too, because they always make it sound like, as you said, a Franken virus. But if you think about, like, there's Frankenstein's monster, right? And he's scary and bad. But then there's also, like, Frankenweenie. And this, to me, is more like Frankenweenie. Correct. It's just like saying, like, okay, we can stick a bunch of stuff on here, and now it's going to be immediately juiced up and so scary. And all I have to say to that is, why don't you try cloning a virus? Try cloning something easier than a coronavirus. Try making a pseudovirus, actually. Yeah. Try making a VSV pseudovirus and get back to me if that works for you. Because I strongly suspect that none of these people could probably even pop GFP into a standard expression plasmid, much less clone a coronavirus and make some sort of, like, super coronavirus out of it, just by, like, I don't know, thinking about ways they could make viruses worse. I wish that I had the skills to, like, have enough foresight to do molecular virology like that. Just, it would make my experiments a lot easier and it would save me a lot of cloning time. But, unfortunately, we're not that good yet.
Infectious experiments. So as well as the benefit being small, according to Rambo, the risk is suddenly sounding like it might be quite high. Is the risk-benefit ratio really right here? A few weeks later, still not knowing that a pandemic was brewing in Wuhan, Peter Daszak gave an interview to Vincent Racaniello in which he explained in more detail what they were doing in the lab. Coronaviruses are pretty good. You can manipulate in the lab pretty easily. The spike protein drives a lot about what happens. You can get the sequence. You can build the protein. We work with Ralph Barrick at the University of North Carolina to do this, insert into the backbone of another virus and do some work in a lab. So if you're detecting viruses,
basic biological research, right? Unrelated to SARS-CoV-2.
This actually makes so little sense that he is just describing essentially Vincent's a virologist and Peter's not a virologist, actually, but he was the leader of some of this work, doing this virus discovery, doing virus ecology, essentially. They are talking about probably the most basic experiment that you would do with any kind of virus, like just trying to characterize it. Can it get into a cell? Can it replicate in the cell? All this stuff. It is not mysterious. And importantly, they didn't know what SARS coronavirus 2 was when they had this conversation. Just like you didn't know what it was, Christian, when you applied for the Creed grant, just like none of us knew what it was before it happened. They talk about this. They frame every single one of these little suspicious pieces of evidence that they want to share with us as something that we were covering up, rather than the fact is, just like everybody else, we didn't know about this virus either. We just knew about it when it caused the pandemic.
If you're detecting viruses in bats, but you can't grow those viruses because they're not in a good enough state to get them into the lab and grow them, then you can at least extract their sequences and from that synthesize their spike gene and stick the spike gene like a cassette into another virus that you can grow, and then you've got a chimeric virus that you can test on cells in the lab. Now, Ralph Barrick had invented these techniques and had collaborated with the Wuhan Institute of Virology to do this work. It would be nice to know what he knows. The University of North Carolina has steadfastly refused to share his emails and is fighting in the courts over that. And he himself has not given any evidence to Congress in open session. But he has gone on the record to say of the lab leak as a theory
in Congress. He hasn't been called, first of all, to Congress, and I'm sure if he was, he would give evidence. But second of all, why would he just give evidence to Matt Ridley when Matt Ridley has repeatedly accused him sort of being the guy who started the pandemic. I don't understand any of this, that Ralph is suddenly very suspicious because he hasn't said anything when these people have been saying for the last 6 years that he started this.
No, and to be clear, Ralph Baric gave a, I believe, 8-hour transcribed interview, right, where Ralph talks about all of this, to be clear. So if you want to hear from Ralph, it's all there. As per Ralph's emails, I believe tens of thousands of his emails have been released as part of FOIA requests, right? So again, this is just not correct.
Yeah, it's completely incorrect. And the idea that Ralph is some sort of mystery person who nobody's heard of. I mean, literally everybody in virology knows who Ralph Baric is, and most of us have at least talked to him, if not published with him.
Correct.
Go ahead, Gigi. Sorry, did you have something to add?
No, go ahead. Let's start
There's a theory about the origin of COVID. You can't rule that out.
Correct.
You do know what some of these experiments achieved. This is a tardy report from the eHealth Alliance that was eventually filed, which shows one experiment in this slide where they took the spike gene from a virus called SHC14 and put it into the backbone of a virus called WIV1, and the result was a 3-fold increase in the death rate of mice and a 10,000-fold increase in the viral load in brains and lungs.
Okay, now time for me to talk about one of the things that really pisses me off almost more than anything else in this whole discussion. The idea that an increase in viral load, which in this experiment, I'm pretty sure is measured by RNA, which means they're measuring the viral genome copying itself, but they're not actually measuring the amount of infectious virus that's being produced. They are saying that because there was a 10,000-fold increase in viral RNA in transgenic mice, these are not humanized other than they express the receptor for this particular virus, the human receptor. The virus grows more. It's a mouse. What does that tell us? Nothing. It doesn't tell us anything about how much sicker that virus would make a human. It doesn't tell us anything about how much more virus would be shed from an infected human. It doesn't even tell us how much more virus is being shed from an infected mouse. So the idea that this is a dangerous gain-of-function experiment that would result in a virus that would be more infectious or more pathogenic or more transmissible in humans is just completely off base, because that's not what this data shows.
Yeah, and to be clear, like that 10,000-fold is during one part of it, like towards the end, it's virtually the same. And importantly, it has no relevance to SARS-CoV-2 whatsoever, right? These are far away from SARS-CoV-2. These are SARS-1-like type of viruses. Again, completely irrelevant as we're talking about the origin of SARS-CoV-2 itself.
Exactly.
They put it into the backbone of a virus called WIV1, and the result was a 3-fold increase in the death rate of mice and a 10,000-fold increase in the viral load in brains and lungs of humanized mice.
I'm just going to grab some water.
Gain of function.
If you need to talk loudly, and I'll be right back to pause it.
Dodging that. Of course it is gain of function. But
when they talk about gain of function, what they really mean is magic wand. That is really what they mean. They don't have any idea of what is happening here, but that's what they're communicating to the audience. It's just a magic wand.
Yeah.
However, what other viruses were they doing these experiments on? And the answer is we don't know. We know which viruses they were working on up until 2016, but we do not know which ones they were working on in later 2016,
and we're never going to learn them
18
again.
and 19. So it's possible that they were working on ones that were
and again,
more closely
they have
related
reported on
to
this, right? Like, and again, they're published papers. We know everything they've ever done.
We probably would know if we could see their viral pathogen database, which they maintained and kept open to outsiders up until suddenly they closed it at 2 a.m. on the 12th of September 2019.
Incorrect. So that database, in fact, did not go. It was somewhat intermittently live until February of 2020. But again, also notice the timing here. Now he's mentioning September. So again, he's suggesting that a lab leak happened in September. So just keep that September month in mind, because I'm sure he's going to mention later dates at a later stage. But Angie, you are muted. But yeah, this is, again, just false.
Yeah, I was just going to say that you notice all this date creep, and that goes right along with this whole where they keep implying that basically we knew that SARS-2 existed before the pandemic actually started, which is completely untrue for everybody in the world.
Yeah.
2019, a couple of months before the pandemic started, and they've never shared with it since. The reason they give for not allowing us access to that database, which would exonerate them in a flash if it has no relevant viruses in it, the reason they give is because they don't want people hacking it. But if you share data, you don't have to worry about people hacking it. So I don't understand that excuse.
Let it
This
run.
is an internal database that they used with their collaborators to keep track of all the viruses that they'd found, many of which, as Christian has pointed out numerous times, have been published. So I don't really understand why this is such a suspicious thing. Like, I don't give, random people access to my lab's data drive either. Because
they also are in bad faith. They're not reliable. They're not going to use the database. You would not expect that.
It would not exonerate them, right? I mean, they have already said they didn't have the virus and showed the results where they were testing for it, and it was not there, right? So that should exonerate them, but of course it never does, right? They
it
would
never
just
does.
say, oh, they removed SARS-CoV-2 from the database. But again,
there's always one more piece of imaginary evidence that they need to produce in order.
And again, the database was online intermittently into 2020, so it's a non-starter.
I should add the database is like an Excel spreadsheet too.
Right.
Excuse. We do know, however, from a grant renewal document to the NIH from the EcoHealth Alliance that there was a plan to broaden the range of viruses they looked at, to move away from ones that were very close cousins of SARS to ones that are 10 to 25% different.
Oh my God.
The reason for this is
finding out about new viruses
this project
now.
is going to be to produce a vaccine that will work against
if they
any
had a
kind
coherent
of
story, they would
virus.
present a coherent story.
Now, it's interesting because SARS-CoV-2 is
Well, I like this now because they're going to talk
different
about
in the
developing a
spike gene
universal
from SARS.
coronavirus vaccine,
So this
like
is
it's
exactly
the worst
the kind
thing
of
you could
virus they were
possibly do. Develop a
moving
vaccine
to
that
take
works against all of
seriously
them.
from this point
No.
on.
Don't worry, we
When
won't be
this
doing that
sequence
here.
first appeared,
Yeah.
it alarmed a number of Western
Nice
virologists.
picture.
It's Christian.
Christian Anderson wrote to Anthony Fauci at the urging of Jeremy Farrar on the 31st of January 2020 to say that I should mention that after discussions earlier today, Eddie Holmes, Bob
glad you
Carey,
wore a
Mike
different shirt.
Hazan, and myself all find the genome inconsistent
I was wondering if he was
with
going to do the Superman
expectations
thing.
from evolutionary theory.
Do you just want to?
I should have made a quick little clip of this, but 2 summers ago in 2024, Matt Ridley debated one of me and Christian's co-authors, Stephen Goldstein, and at the debate, he Superman'd open his shirt to show us that he was wearing a Biosafety Now t-shirt that had an alignment of the furin cleavage site and RATG13 and stuff on it. And then Christian's famous quote, that it is not consistent with expectations from evolutionary theory. And that was one of his big owns in the debate. It was a real moment. We got to see Matt Ridley, threaten to take his shirt off. Okay, let's hear what else he has to say about you.
What did he mean by that? He meant that in the middle of the spike gene
That was
of
the shirt.
SARS-CoV-2, there is something that sticks out like a sore thumb. A feature called a furin cleavage site that massively increases the ability of this virus to be infectious in human beings because it enables it to use a human enzyme called furin.
All right, I just have to make one quick statement here. Yes, it's true that SARS coronavirus 2 is the only sarbecovirus we found that has an intact functional furin cleavage site at the S1, S2 junction. However, every single coronavirus that exists requires cleavage at the S1, S2 junction in order to remain infectious and go through its entry and replication cycles. So it's not just furin, it's always host proteases that cleave this. There are a number of other ones that can do it. It's just that this happens to be the only sarbecovirus that uses furin. Again, they all do this at this site. They all cut spike protein at this site. So this actually isn't that weird because many, many other viruses, including flu, including Ebola, including, I'm forgetting, herpes. Some herpes viruses have furin cleavage sites. Like they evolve furin cleavage sites because proteolytic processing or cutting of these proteins is required for many viruses to actually carry out their infectious cycle. So
it also seems like an end problem, though, too. I mean, do we know all of the viruses that are on the planet?
No, we don't. We absolutely don't. And we do know that furin cleavage sites for some viruses, for influenza virus and avian influenza viruses in particular, can lead to a more pathogenic virus. It is linked to pathogenicity in many cases, but sometimes it's not because of furin. Sometimes it's because that is the most efficient way to cleave whatever protein that is of the virus. And if the virus is cleaved more efficiently there, then it can replicate more efficiently. So it becomes more pathogenic because it's basically a better virus and it can grow more. They completely miss all of this point because they're actually not virologists and they don't really understand this. They seem to think that this furin cleavage event for SARS-2 is this magical thing that only happens for SARS-2, when in reality, the thing that's unique about it is it's just the protease furin that's doing it. And these other ones, they've got cathepsins and, all these other cellular proteases that basically do the same thing, because all of these viruses have to cut the spike protein at this point.
Yeah, and I think to the furin cleavage site, I do because it's true that we haven't found a SARS, a virus that has that, right? But I think we probably will. Like, again, the question of enough sampling, right? But one thing, Angie, I just want to mention, because he does mention my email there, right, where I do say that we find the genome inconsistent with expectations from evolutionary theory. But just to be clear, in the very same email, which he does not mention, is that, but we have to look at this much more closely, and there are still further analyses to be done, so those opinions could change. Is it literally the next sentence right after what he mentioned there? But he, of course, took that out.
Yeah, and if we have time at the end, and I'm starting to wonder if we will, just because there's so much to unpack here, but you've brought in copies of a lot of your papers, and I think this is a consistent pattern that we see, where we see the first part of what you say gets mentioned, and then the part after that that's putting it into the proper context, where we're saying we don't know everything yet. This is unusual and we're concerned about it, but we're going to look into it more and figure out what's going on based on the evidence that we find, not just on like, oh, furin cleavage site, somebody must have put it there. Which virologist was it?
Yeah.
A human enzyme called furin to cleave the spike and facilitate entry into and out of cells. But what
he doesn't know
what this is not a mutation. No. It is an addition. It is an insertion. 12 extra nucleotides, 4 extra amino acids have been added, and this is clear by aligning the sequences with other viruses. This is the first and only discovery of a furin cleavage site in a Sarbeco virus ever made, out of many hundreds. It's quite striking.
Well, we won't find any more because you guys are going to shut
down. We'll find more.
Everybody who saw it.
We'll make a bet.
But when Xi Zhengli published the sequence in Nature, she didn't mention it. She doesn't use the words furin or furin cleavage site in the paper at all.
Okay, so I'm not sure if this has ever been answered, Christian, if anybody's asked Zhengli about this, but I'm pretty sure, like, at least we all just decided, and I'm curious to know if we have any evidence to support this, that the main reason that furin cleavage site was not mentioned in this was because this figure that he's showing right here specifically concerned the S1 portion of spike. Right. And so why would you mention the furin cleavage site if you're not concerned about the cleavage event? You're more concerned about the receptor binding domain, which is what this was about. Right. I don't see that there was any reason to mention the furin cleavage site, so I don't understand why it's so suspicious that they didn't.
Yeah, and she has answered that question. She just missed it, right? And to be clear, most people did, right? Yeah. The vast majority of scientists completely missed that this had a furin cleavage site in there. And we're talking early 2020, right? She's extremely busy working on a lot of different things. She's not a furin cleavage site expert. So she just, guaranteed.
Well, she's a coronavirus expert, and guess what? There's a coronavirus outbreak in Wuhan, the city that she lives in. So I'm assuming that she was working on that.
Like suggesting there's something nefarious because she doesn't show her furin cleavage site in a supplementary figure, but at the same time she published the sequence of the virus she had just sequenced. I mean, it's just ludicrous. Like it's simply just ludicrous.
It's so absurd.
Yeah.
Oh. And her diagram of the first half of the spike gene stops just before it would appear.
Right, that's the S1, S2 junction, and that's
this
where
is like
the S1 portion ends.
And not mentioning the horn.
Oh, this fucking
that diagram stops
thing.
there.
Sorry, freaking unicorn.
But the end of the S1 section of the spike gene is there.
I've had enough of this too. This is a common thing that Ridley and his co-author Alina Chan frequently say, that SARS coronavirus 2 is like if you found a horse with a horn on its head and you didn't think it was a unicorn. But the fact is, okay, so it's a horse that has a horn on its head. You know what? There are deer that have horns on their heads, like that actually look like horns and not antlers. I didn't know this until I was actually looking up some of the species that were at the Huanan market. So there's a lot of different animals that have a lot of different features, and viruses are no different than this. Just because you find a virus that has a unique feature on it, like a furin cleavage site, does not mean it's a unicorn. It means it's like a brunette. I mean, this is ridiculous. We keep talking about, Matt keeps talking about SARS coronavirus 2 as though it's this magical thing that we've never seen before. But the reality is we just haven't seen a virus like this particular one before. And every single virus is unique. SARS coronavirus 2 is different from SARS coronavirus 1. They're both unique viruses. These are the differences that we're talking about here, not some sort of like big secret that we found this unicorn like super virus and then everybody tried to bury it or else make it worse.
No, but I think like one key thing here too, Angie, which goes, you know, as we're talking about this, generally speaking, right, is just the fact that, you know, SARS-CoV-2 is that virus that did cause a pandemic, right? So because, and SARS-CoV-1, for example, did not. And that means that SARS-CoV-2 is unique, right? Because if it wasn't, it wouldn't have caused a pandemic in the first place. And is the furin cleavage site a key component of what made SARS-CoV-2 a pandemic virus? Probably it is, right? But really importantly, is not the only feature. It's the whole virus with the furin cleavage site being part of that. But that's really important, right? Because the survivorship bias, we are looking at this virus specifically because it caused a pandemic. And because it caused a pandemic, it's got to be different than everything else that's out there. It's simply as simple as that.
Exactly right. Couldn't have said it better myself.
The spike gene is there. Why not go on to the S1, S2 boundary? And as you can see, the furin cleavage site has 2 arginine residues in it. That's why it's good at attracting
See, he does not think that anyone actually is looking at the arginine residues. It's just all to make him sound like he knows what he's talking about. He doesn't.
Also, this is probably the worst.
You don't need to, you can keep it going.
Yeah, let him see what he says next about CGG.
Well, that's unusual, because these kind of viruses do not use CGG for arginine very often at all. It's the rarest codon to use. And yet here are 2 of them next to each other. And this is the only occurrence of 2 CGG codons next to each other anywhere in any sarbecovirus genome that we've found so far.
It means Zhengli Shi was here, according to him. No, seriously. I mean, this is why he's talking about these rare arginine codons. Is basically, it's like some molecular virologist like signed it. It's like Picasso, except it's the furin cleavage site at the S1, S2 junction in the spike protein.
Yeah, and to be clear, David Baltimore actually retracted that comment that there was the smoking gun, right? And just as background, in SARS-1, for example, about 5% of arginine codons are CGG. In SARS-CoV-2, it's about 3%. So there is really no big difference there, but we'll get into this more in the end.
Virus genome that we've found so far. No wonder David Baltimore, when he first saw this, said this could be the smoking gun. He later wrote back from that under pressure from colleagues, but that's what his first thought.
Oh, right, because we are very good at putting pressure on like
a wasn't interested in
Nobel Prize winner.
Maybe that wasn't her thing. I'm afraid it was. She'd spent a lot of time, as had Ralph Barrick in the months and years leading up to the pandemic, interested in how much furin cleavage could enable you to grow viruses that you couldn't otherwise grow, and was a limiting step in how well viruses infected. Here is a paper that she published in 2017 on a rodent virus where she draws attention to a very similar furin.
Did she publish this all by herself?
Here's a paper published by her colleague Shibo Jiang.
I know, but the way that they focus on her is just very sexist.
A furin cleavage site onto a gene, albeit not a viral
Although
gene,
I'm
but
sure
an
if you
antibody
said
gene.
that to
A
the
12
Viscount, you
nucleotide
would probably point out
sequence.
another slide in which he also circled.
And here's a
Dr.
virus that they
Ralph
do
Barrett's
mention
name.
in their Nature paper, a cat virus, coronavirus, which has exactly the same furin cleavage site. So they would know that the sequence PRRAR.
I wish they'd show the audience.
Everybody already knows that PRRAR can work as a cleavage site.
Look at the nucleotides here. They're completely different.
Yeah.
And that letter from Christian Andersen to Anthony Fauci resulted in a flurry of activity. For a while, we weren't able to read the emails, but then we did. And this is what Fauci said to Farah. I just got off the phone with Christian Andersen. He related to me his concern about the furin site mutation in the spike protein of the currently circulating virus. I told him the sooner
Anderson the Swedish way.
Examine the data and, if necessary, report it to the FBI and MI5. Quite right, too. That's the right response.
Clearly, Fauci wasn't trying to cover it up. He talks about reporting it to the FBI and CIA.
Collins.
I'm not clear where you guys went wrong, because Ridley's like, quite right.
And the upshot of that meeting was that they agreed that they would write something about this, and that Anderson would be tasked with drafting it. And after the meeting, Farah and Anderson and Holmes all still thought it was quite likely as a hypothesis that the furin cleavage site had been inserted. But sometime over the next few days, they changed the line they were going to take in this article they were drafting, and eventually ended up writing something that ruled out a lab operation.
Well, I'm pretty sure it didn't rule it out. It just said that it wasn't likely.
So you want to pause here. So just to be clear, so you know, again, we discussed many versions of a potential lab leak, as I mentioned in the beginning, right? Like, one thing is inserting the furin cleavage site, is one is passaging SARS-CoV-2, for example. And at that time, like, we were sort of like, oh, actually the insertion doesn't make sense, but the passaging I was still pretty focused on. The idea that we agreed that we would write a paper, and that is not actually true, right? We agreed we would write a summary of what was discussed. And then over time, and in our paper we completely dismissed, is not correct. In fact, our hypothesis 3 is selection during passage. So all of it is in there. We'll pull up the paper once we are done with all of this, and then we discuss it more.
Yeah, that sounds great.
Record is saying that he'd had a good look at RATG13 for the first time, and this convinced him it was different. But
that is not correct.
Plausible.
I mean, I can't believe that you were like, look at this RATG13. How this would actually work in practice. And I'm actually so glad we're discussing it here, because some of this stuff is so stupid. It's basically imagining what's going on in your mind and recording it as if it's fact.
Yeah. And can we just for a moment, you know, again, we are actually talking about the first speaker of an inaugural lecture series here, giving this talk at the NIH right now, right? And he's talking about the former NIH Director, Dr. Fauci, as well as scientists discussing here what may or may not be true. It's pretty remarkable to see that. And not yet, I mean, I'm still, you know, looking for that one data point that he's actually showing, like, this is why we know it's a lab leak, right? And a hypothesis that goes with that. We haven't gotten to that either yet. But let's see where it goes next.
I fear we're going to still be waiting indefinitely, Christian, forever.
I think so. Yeah.
But that's not really plausible, because he had been tweeting about RATG13 a week before. True, by the end of that week, there was indeed a possible excuse for why they could start to rule out a lab leak, and that was the news from Jeremy Farah to Francis Collins, that reports were coming out of a 99% similar virus in a pangolin, the missing link. And Collins quite rightly replied, very interesting, does it have the furin cleavage site? And the answer was no, it does not.
Yeah, and it was not.
Is it 99%
it's totally irrelevant.
So
sequence,
it's only 90% similar. It's not.
I remember talking about these pangolin viruses too at the time. It was a big deal because it had just come out and none of us knew anything about this new virus.
it's important to note that the effect of this announcement was to throw a lot of us off the scent for a while. Whether that was its purpose, I can't say.
Why was he on the scent?
But as
what scent was there in February?
Paper, ruling out
of 2020,
Christian Andersen knew that the pangolins didn't help. Unfortunately, pangolins don't help clarify the story.
No, because they're a distraction, essentially. We didn't know anything at the time, and it ended up not being relevant, but we thought at first it might be.
SARS-CoV-2, and it said, we do not believe that any type of laboratory-based scenario is plausible. And in a press release from Scripps, Andersen went further and said it rules out laboratory manipulation. Now, I was misled by this paper. This was what led me to go and say to colleagues who were asking about this in Parliament in the UK, no, it's been ruled out. It wasn't a lab leak. There's good evidence it's been ruled out. But I hadn't read the paper very carefully. When I did, I found the arguments rather flimsy. I mean, the main argument was that the fit of the receptor binding domain to the human ACE2 receptor
what
was
his
good,
colleagues in Parliament
perfect.
In the House of Lords, that would be a bunch of other people like him who are
that's a very
born into
rational
it,
argument.
Trying to figure out whether it was a
only
lab leak.
Many months later that we found out that Christian Andersen had not changed his mind.
He did.
Oh, he had. That's why he wrote a paper.
He said on the 17th of March, we do not believe any type of laboratory-based scenario is plausible. On the 1st of February, he thought it was frigging likely.
The 1st of February.
2nd of February, after the meeting, he still thought natural or escape.
A month and a half prior to that paper.
And on the 16th of April, a month after publishing the paper, he's still not fully convinced, can't fully rule it out.
You and
Andrew both correctly said you can't fully rule anything out.
Can you ever scientific paper rule anything that says
out?
And you just paused it there.
What you think in private is.
Yeah. So to be clear, so he is obviously, so the first is early February. Again, I believed it was a lab leak, and in fact, I did for much of February, as I've explained many times, right? Again, initially I thought it was engineered, right? But that was for a split second. Then so that particular manipulation was off the table, but the passaging idea, which we also describe in proximal origin, right, was on the table. Now, to be clear, he's talking about April here, and at that time, the Washington Post had reported there were secret cables out of the Wuhan Institute of Virology, which showed that there had been, unsafe practices and there had been accidents and things like that. And that is what I'm reacting to here in private messaging among, scientists here, where I say like, well, wait a minute. Might we actually have been wrong in our earlier conclusions, which is exactly what I'm saying here, right? But of course, what we realized was that those cables were not actually anything to do with unsafe practices or lab leaks at the Wuhan Institute of Virology, right? It just had to do with very basic maintenance of what happens in virology labs. So, but again, it's a good example of like, have I questioned our own conclusions over time? Of course I have. But as the continuation of the science clearly show is that those are split seconds where I'm like, let's just go back and just make sure we did that right or wrong. Think about it, do more analyses, look at more data, and then at that point you're like, you know what? Actually, our earlier conclusions hold up, and of course those are later papers.
Christian, it's almost like you're talking like a scientist or something where you're
it's weird, right?
Not going to be convinced even by your own data, and you're going to go back and just confirm that, yeah, I did that right. And in fact, the lab leak people, including Ridley, love to make a big deal about the fact that we've put a correction on the Warby paper, and also you guys have put a correction on the Pekar paper that I wasn't a co-author on, because we got minor things wrong in these papers when we published them. They don't change any of the data, they don't change the analysis, and they don't change the conclusions, but we made errors, so we corrected them. Yeah. That is a normal part of the scientific process. And what I don't understand with a lot of these conversations is how, on one hand, you are supposed to be constantly questioning yourself. But on the other hand, if you do question yourself and then you change your mind based on the evidence, you're lying.
You think in private is not good scientific practice.
Yeah, and just pause again, because this needs to be very clear, right? what he's presenting here is as if I said one thing in private and one thing in public, which is just flat out false, right? Again, I have been very open over the years about how I initially believed it was a lab leak, including at the time when things like Twitter, for example, was being discussed. And this idea that there will be one thing in private and one thing in public is just completely false, right? And again, of course, what we do is present our conclusions and analyses in scientific papers. And if people don't agree with that, that's up to them, right? But of course, that is about as public as you can be, including your entire reasoning behind why you will conclude one way or the other.
Yeah. And, I think the basis for saying that you thought something else in private is ridiculous, too, because really this comes from conversations that you were having with our other co-authors on the Slack channel.
Yeah.
And unfortunately, I mean, this type of thing, this has had such a chilling effect that we've all had to change the way that we communicate with each other, because we don't even feel safe having, just like we're shooting the shit like friends and colleagues normally do. Which is when I first read your Slack messages, and I didn't know you as well then as I do now, but when I first read them, they looked like any kind of Slack messages I could have sent to my collaborators and friends. They look like any number of text messages, like text messaging things that I've had with people. Because when we're with our colleagues and friends who we work with, we tend to make jokes. We tend to talk about things that are meant to be private, because we're not maybe sure like where things stand in our heads. And that's what I saw a lot of with you guys too. You were actually trying to work out the science. And so you were exploring a number of these different hypotheses. That's not the same as like saying, I commit to believing in this hypothesis forever, and now this is my faith.
Yeah, and again, all the context is fully stripped here. Right?
Yeah, you were just exploring all of the different hypotheses, which is a fundamental part of the scientific method.
Including questioning your own prior work.
Exactly. Especially questioning your own prior work.
Written to the editor of Nature Medicine, requesting that this article be retracted or corrected, have not even had the courtesy of a reply.
Well, I mean, let me just say really quick, maybe he didn't have the courtesy of a reply because you got a bunch of lab leak crazy people together and turned it into this whole big performative stunt where you brought a manila envelope to literally the offices of Nature Medicine here in New York City and made this big spectacle about delivering this demand for retraction. Okay, like that is just comic. It is a stunt. It is not a serious thing. And I would expect the editor of Nature Medicine to completely ignore that because it's unprofessional and ridiculous.
Reply. We do have quite a good explanation of what had caused them to change their line, if not their mind. And it comes in an exchange between Andrew Rambo and Christian Andersen. Given the shit show that would happen if anyone seriously accused the Chinese of even accidental release, my feeling is we should say that given there's no evidence of a specifically engineered virus, we cannot possibly distinguish between natural evolution and
testimony that Christian gave with Bob Gary to the COVID-19 Select Subcommittee in 2023, because they had Marjorie Taylor Greene, I believe, had a big blow up of this quote from Andrew.
It's impossible not to.
They really had an issue with the fact that Andrew mentioned politics at all.
The decision to write the paper the way they did was political rather than scientific.
So just to pause again. So again, of course, what we show like is that, of course, our conclusions are scientific, but we also have to realize that whatever we write, a lot of people will read that, including politicians. So let's make sure that when we say something, we back it up with evidence, we write it clearly, and we try and make sure that we're not just writing this to a scientific audience, but to a broader audience. That's exactly what that is. And given where we are today, I think Andrew might have been right.
I think Andrew was completely right. And I mean, there is no way. And I really want everybody who's watching this to listen in, because I think that this is one of the things that's really harming us in this fight that we are engaged in for basically the future of our profession and our country. And that is the fact that, when scientists like to stay away from politics, they don't like to get political, because we are used to not worrying about the politics. We're used to worrying about the data. But the fact is, all science exists within the context of society, and part of that is political. And now it's more political than ever. And I think it is really important for scientists to think about the broader impacts of their work, whether that be social, cultural, or political impacts. I think Andrew was absolutely right on. You don't want to say, even if that's what the science suggests, that, oh, this was a lab leak from China, unless you have really solid evidence to back that up, because that's going to have huge political implications, even if you don't care about them. So I think this is the responsible way to do science, is to understand that it does happen within the context of a political society.
Yeah, and I think in general too is that, like, whatever you say, make sure that you actually do base that in some level of evidence and reasoning, right? And again, that is exactly what Andrew is referring to.
Exactly.
In September 2021, a document was leaked to the press that should have been made available from the very start.
No, it shouldn't.
Diffuse proposal. Lots of people knew about its existence and chose not to tell us about it. It's a very important document.
It was an unfunded DARPA proposal.
From Peter Daszak.
And in fact, Dr. Barrick was asked about this, and he said, you know what, I'd forgotten about diffuse.
You know, most of us, I don't know about...
I write a lot of grants, I forgot about it.
I don't know about either of you, but when I submit a grant, I tend to put it out of my mind unless I'm awarded that grant. Yeah. I have other grants to write and I have other projects to work on. So once it's in there, if I don't need to be actively thinking about it, I don't until I get it again. And then if I don't get the grant, and I should add, I was a co-PI on a competing grant that was also not funded under the same exact DARPA program. I had to go back when diffuse came out and look at what our grant proposal said to make sure that people weren't going to start digging through the archives and trying to take what we said out of context. We weren't doing any coronavirus work. But, I don't think about grants that I have applied for that I don't get. And I don't just go digging through the couch cushions looking for a spare $18 million or whatever to do the work anyways, if I don't get it funded. If I had the money to do the experiments, I wouldn't take all the time to write the grants. And anybody who's written a DARPA grant knows that that is a special form of hell that you would not absolutely do under any circumstances if you already had the money to do the work. So the idea that this should have been released immediately because this unfunded grant holds the secret to like secret coronavirus research that was happening anywhere. No, why would they have applied for this dumb grant if they had the money to do the work already? They could have had Ralph just like already making all these coronaviruses.
Well, this is important because what he's doing is showing, like, he's taking things that are normal and normal part of doing science. He's communicating it to people who don't know the way that science is done, and he's making it sound suspicious. This is what he did with HIV, AIDS, and the polio virus. Trying to say that that came from a polio vaccine manufacturing thing. It's what people did. I mean, they do this every time. They just take things that are normal, they other it, and then they proceed like it's a big conspiracy. I was told after the anthrax letter attacks, people were making it a big deal out of the fact that like some people work late into the evening at UCM, and it's like I was a postdoc. I thought that was my job to work all the time, because it was my job to work all the time, but they just didn't know what the way that science is done, and so they make it seem really weird. Sorry, you can keep going.
I'm not going to tell anybody about those like midnight time points I took on all my grad school growth curves. So just really quick, a little housekeeping stuff. It's 4:00 right now, which is when we're going to start the after show. And I underestimated clearly how much pausing we would need to do. I think we should just hit start. I'm going to bring our colleague Anna Culbertson in onto the stage with us. And Anna, if you don't mind, we'll introduce you really quick, but then we're gonna, I'm sorry, I keep muting you. It's like an ASIP meeting in here. Go ahead and unmute yourself because I feel like I keep trying to unmute you and you keep trying to unmute yourself. And, oh no, you can't unmute?
No, you're good. Hi, I'm Anna Culbertson. I'm the executive director, co-founder 27 United, an NIH Alumni Action Network, trying to deal with this. So here in time, happy to talk and just kind of talk more about what's going on in the current moment in time with NIH and just kind of like what this means in the broader context of like the attacks really on our academic institutions. But yeah, happy to just keep going through this with you because this is such an interesting dialogue. It's really interesting to hear from all of you who basically are like, no, this isn't what I was talking about. Like, it's crazy to just see them misquote you, like you in real time, Christian. And like, it's just so, from an outside perspective, this feels very meta and just like really kind of like out of this world. So very, very cool to be a part of this experience.
Well, I'm really glad we did this because of that exact reason. I think that your colleagues at NIH, or your former colleagues at NIH, because I should have mentioned that Anna is a former program officer at NIAID, who was let go, unfortunately, because, you know, they're firing everybody unfairly and unjustly. And Anna, to her great credit, has turned that into organizing and advocating for our federal colleagues in public service. And so while she's not an expert on this topic, that's what you just said, Anna, is exactly why we wanted to do this. And we think it's important because this has been going on and it has had a profound effect, I think, on all of our lives and our mental health. At least it has on mine. I can't even imagine how much more difficult it has been for Christian. But, you know, we have really never had the opportunity to respond to this because the only types of discourse they want to have with us is either a debate where sort of they control everything and it would be very difficult to actually have a reasonable scientific discussion. Or, you know, they're having like these sort of lab leak events like this that we're just completely excluded from. And so we just look like these people who don't want to defend ourselves when the reality is like there's not a lot of good forums for us to defend ourselves in and for us to actually confront these people about these things because they tend to not, you know, engage us in good faith. So I'm really, really glad we did this. And I think I'll just start this. And why don't we just keep going until we're done? And then people can join and you know, everybody can watch however long this thing is. Once it's finally over, it will be on my Substack and Twitter and on YouTube. And so everybody can share it and watch it at their leisure. All right, let's get started again.
Virology and a number of other groups for $14 million from DARPA to investigate defusing the threat of bat-borne coronaviruses. And it stated halfway through the text that they were interested in furin cleavage sites and that they were interested, if they couldn't find one, in putting one into a sarbecovirus for the first time.
Now, I'm just going to add some little inside baseball here about a DARPA grant. Where they got this information was from something that's called the statement of work in the DARPA grant. Statement of work is like a big long list of all the things that you're going to do in a grant. You normally don't have to do something like this for an NIH grant, for example. But this is like literally a list of tasks and outcomes that will come from this and milestones. And so when DARPA funds your grant, they don't actually fund it right away. They will say, we're going to fund it, but now we're going to engage in contract discussions with you. And during those contract discussions, they will go over your statement of work line by line, and they will take out things that they don't think are priorities for the program. Also, once you're doing the work, unlike an NIH grant where you just have to submit a progress report every year, for DARPA, you usually get evaluated monthly. So if you're not meeting your milestones, you're not meeting your deliverables, they will actually cancel your grant. So having that one thing about inserting furin cleavage sites into sarbecoviruses, it was one item on a statement of work. It was not the basis of the entire grant. It was one thing that they were going to do among several others to characterize these novel viruses that they found to determine which ones might actually pose a threat to people. So I just want everybody to know that when he's reading this, and you can actually even see, oh my God, in this bottom quote, the first words are cathepsin L. You know what that means? They were trying in this thing in the statement of work to determine which protease was making a cleavage at that site, which one would be most efficient to make a pathogen. They were trying to understand the cleavage process better. They were not trying to juice up viruses with furin cleavage sites. They wanted to see what kind of effect that would have relative to other proteases that do the same thing and how that would impact these viruses' potential as a pathogen, which again, we know that there are thousands, millions probably of these viruses. How do we know which ones we should worry about, which ones we should devote our resources to? It's by doing experiments like this. It is not just oh, we're going to stick some furin cleavage sites in and see what happens, it'll be fun, that he's presenting here.
Time. We will introduce appropriate human-specific cleavage sites and evaluate growth potential in monkey cells and human airway epithelial cultures. Now, some people have said, oh no, they were talking about putting it in at the S2 site, not at the S1, S2 junction. No, the diagram makes quite clear that they're interested in both sites.
We don't have to pause here. Let's just play the graph, but we can bring this up afterwards.
Yeah.
This is a deep misrepresentation, right? And we can take
Xi
as part of the grant. We have
note that although he says the work will be done in North Carolina,
if there's a person that's not totally asleep in the audience, I will be shocked.
He really is going to talk for an hour and a half.
The biosafety level 2 nature of the work on SARS-CoV in Wuhan makes our system highly cost effective. Much cheaper to do it in Wuhan.
Well, if you're using pseudoviruses, you only need BSL-2. And
China might be
even if you're doing some of this work, I wouldn't do it at BSL-2 with the Bacillus virus, but technically, that's the standard currently in the U.S.
That is the standard, yeah. And Barrick, again, actually explains that in his testimony, right?
Passage in ACE2 transgenic mice.
Barrick chooses to do it at BSL-3, as would I, as would many people.
And see, now he's going back to the email. And actually, this is something Collins is wrong about. They didn't actually do the animal work in BSL-2, right? They misunderstood that.
Let me remind you of the timeline again, of all the gin joints in all the towns in all the world.
Oh, for crying out loud.
One city
actually,
where this kind of research is happening.
FFS.
The year after, someone has proposed to put a furin cleavage site into a sarbecovirus for the very
This is like the that conspiracy where all the things are, the meme with all the things that are tied together. That's all this is.
There's a mutation in the human version of the virus that happened very early in the pandemic. It stabilizes furin cleavage. It's a response to furin cleavage. It implies that the furin cleavage
site response to furin cleavage.
Not been in that virus forever. Chinese government banned the sale of ex-laboratory animals in markets during 2020. 3 key lab workers, according to US intelligence, I can't verify whether this is true or not, fell ill. They included Ben Hu, who was in charge of these chimeric virus experiments, and Ping Yu, who, as we saw, was the
it goes without saying that not only can he not prove
the man
it,
who developed a COVID vaccine very early in the pandemic in China died reportedly by falling off a roof.
Oh boy.
To summarize, ladies and gentlemen, in 2017-18, in Wuhan and nowhere else, the Wuhan Institute of Virology shifted their priority to 20% divergent viruses from SARS.
I just love how it's like the whole institute shifted their priority for
site into a sarbecovirus for the
He's going to expand to more divergent viruses, and so therefore it's like the new institute
at a low biosafety level. And the following years in Wuhan, a lineage 4 sarbecovirus appeared with a newly acquired furin cleavage site in the very same city, leaving no trace in any animal in the markets or anywhere else. And the Wuhan Institute of Virology misled the world, whether intentionally or not, about the name and date of sequencing of certain relevant viruses and took their database offline.
All right, I just have one thing. I can't wait till the end to say something about this low biosafety level stuff. So as we were just saying, in the BMBL, which is the US standard, working with sarbecoviruses that are not SARS-2 or SARS-1, you can do it at BSL-2. That is the US standard. Most people choose to do it at a higher biosafety level or at a higher level of containment, but you don't have to. And
I think, Andy, I'm not sure, you say most people choose, I think you mean Dr. Barrick's laboratory does that at BSL-3. I don't know that that's generally true, though.
Oh, I think it is generally true. I mean, I do. Everybody at my institute does. Everybody that I've spoken with who does do this type of work. I don't do it very much, but people who do work with these viruses, I just work with all sarbecoviruses and all myrbecoviruses in BSL-3. That to me is just the easiest way to do it. Also, that way I don't have to have a bunch of viruses in my BSL-2 lab and a bunch of them in the 3 lab. Like, it just makes sense to me to take those extra precautionary steps. But that said, you can work with respiratory viruses at BSL-2 and never get a lab infection. I did my PhD on rhinovirus. I worked with multiple infectious respiratory viruses that I am not immunized against because no immunization exists. And as far as I know, I never got a lab-acquired infection. I never had symptoms of a lab-acquired infection. I've worked with seasonal influenza viruses that you work with at BSL-2. Those are human pathogens. I have been immunized against them, but not all of the ones. I'm certainly not immunized against like PR8 and WSN and some of these older seasonal isolates. But that said, they're still not very dangerous for me. And I can work with them in BSL-2 because I'm also a very experienced virologist at actually doing laboratory virology work. And I know how to not infect myself with them. And I know how to train my staff to make sure they don't infect themselves with them. And we have biosafety procedures and we have everything. So this low biosafety level is biosafety level 2. That is not a low biosafety level. That is a standard biosafety level. And it does not mean that we're walking around in like a big cloud of viruses. We still use biosafety cabinet. We still sometimes use respiratory protection. We still use a lot of different ways, redundant ways, to make sure that we're not being exposed to these viruses in the laboratory. And to have this little pheasant mating guy like saying what is a good biosafety practice and what's not, I find completely offensive because I have been training to work in these high containment labs for literally decades. I have to have a security clearance to do it. And it's completely unacceptable for this like British dude to just pop off the plane and start talking about how people can't work with viruses safely in biosafety level 2 laboratories.
Well, they're not interested in how things actually work. They're interested in creating this narrative.
Right.
Yeah, that's
actually, yeah. Angie, I think he's almost done. So give me just, I'm just looking
let's finish it up and then we'll all have
let's finish up and have some general discussion here because otherwise we're going to get sick then.
Sounds good.
Now, I'm often asked, if you think all this is important, why haven't you published it in the peer-reviewed literature? Believe me, we've tried. We've submitted papers saying all of this again and again. They're always turned down by gatekeeping referees.
Gatekeepers.
Be able to say you haven't published this in the peer-reviewed literature. That's a circular
argument. No, man.
Sure there's a lot of...
You actually have to make a coherent argument.
I'll end.
There's some predatory journals I'm sure who would love to publish this after peer review.
Who have contributed to this knowledge. Most of it didn't come from me. It came from these people.
Came from Alina.
Thank you very much.
And the Seeker.
What's a clap?
Oh, good.
You paid clappers.
They are going to have a
chat. Thank you for that lecture. We're going to make room for questions, and those of you who'd like to ask questions, please line up at the mics provided. But I thought I would take the opportunity just to get to ask you the first question, if you don't mind. And that has to do with the stakes of the debate. I mean, I have a position, and I think largely agrees with your position, but I know there are scientists who disagree with both of us.
Well, there sure are.
How much does it matter who's right in this debate? Because
not at all, because we're going to shit can the NIH anyway today.
Such difficulty getting a scientific debate going on is because people think the stakes of this debate are so high.
Got to tune up for a few minutes.
That if you're right, then we should not be doing virology at all. That's one version of the I think the imagined stakes. But in fact,
correction,
it seems to
that
me
is
the
their
stakes are
version
not
of it.
Quite that high, right? If the stakes could be that we just do virology much more carefully. We ban dangerous gain of function, but we allow many other parts of virology to continue, which is an essential part of scientific investigation into human disease. So I would love to hear your thoughts about what the stakes actually are in this debate.
Well, I think quite a lot of scientists feel that they have to do their best to resist this conclusion because the result could be a loss of public license for science altogether, which would be catastrophic.
The person who believed it.
Sorry.
Like, no, just Christian, he's here. He admitted it in the beginning. And now it's like, oh, well, he doesn't believe in any, like what you were saying earlier. It just feels so crazy to me that he's like, well, scientists don't want to say that. It's like, well, they already did. Experts already said that this could be true.
And it has nothing to do with the facts. It has nothing to do with it. This is bad faith arguing.
It's called lying. is what it is.
Yeah, it is.
It's known as lying. And nobody wants to actually say this out loud, but I will, because that's what it is. They completely make up what is in our heads, what we are thinking. It is actually incredibly insulting because it strips us of all of our agency. Angie Rasmussen doesn't have an opinion on this. My opinion on this is whatever Matt Ridley or Jay Bhattacharya thinks I'm probably thinking, because, oh, well, I'm just a reckless virologist. I don't want virology to go out of business. How am I supposed to do all of my fun gain of function research if that happens? But the reality is, we have, as Christian said, he admitted everything at the beginning. He was completely transparent to the point of talking about it on Twitter, talking about, this in news articles, publishing papers, scientific papers on it. We've all been, I've been tweeting about everything for the last 6 years and talking to the press about it quite a bit. And so have many of our colleagues. The idea that we somehow are all secretly hiding our deep desire to just save virology at all costs and instinctively respond to the idea of a lab leak with can't let that get out because they might stop us from doing all of our fun, dangerous virology work. Like, that is just quite simply not true. The fact is, if this had been a lab leak, I would want to know that more than anything else, because I don't go out into the field very much, like finding viruses. I did it once, like to see what it was like, and I don't like camping. I like to be in the lab. And you know what happens in the lab? I have to think about safety the entire time that I am in there. I am not thinking, what if there's a lab leak and they might shut down my research? I'm thinking, what if there's a lab leak and like it happens at all? Everything I do and everything I have been trained to do in high containment is to avoid that happening, because it would actually be catastrophic if there was a real lab leak. I would want to prevent that from happening. So I certainly would not want to cover that up, because when there are lab leaks, you analyze what happened so that it doesn't ever happen again. And that is an important and critical part of biosafety practice, which obviously neither of these two know about, because they don't actually know anything about biosafety and they have no experience working in containment. And they, in fact, do want to get rid of all virology research because they want to get rid of all research because they want to give the money to President Trump and undercut the NIH and undercut American democracy.
Catastrophic. And I agree, it would be catastrophic. I think the vast majority of science benefits humankind, and I wouldn't want to see it stopped because of one mistake that might have been made. But I don't think, as you imply, that the stakes are quite that high, or at least were quite that high.
That's what they'd like us to believe.
If at the start they had gone with what Christian Andersen apparently wanted to do, which was say, look, this is a possibility and we need to take it seriously, then I think the damage could have been confined to one small corner of virology, and the rest of biotechnology and the rest of biomedicine and the rest of science would be untainted. But as it is, the way it's gone on these last 6 years, it's now harder and harder to confine the damage to one small set of experiments.
Why?
They're just filled with grievance.
Which, as I say,
yeah, I mean, from all I can tell is just because a bunch of Jews are pissed.
Benefit and relatively large risk. That's, I think, the problem. And I can sympathize with people who think if we admit it now, we're going to lose social license to do science altogether. But I still think that it's vital to, the truth is just too important to take that line.
Well, it's not the truth. We've already
agree with that.
I want to see what other.
Yeah.
The returns to doing science are so tremendously high. We can cure diseases that were, when I was a medical student, I thought would never be cured. Sickle cell anemia, and we can get improvements in cystic fibrosis. We have so many huge investments, many of which come out of this institution that you're speaking at. And so, to me, I would love to see a way to have the truth out, whichever way it ends up. As you know, I'm convinced that your presentation is correct, but others may disagree. But two, to be able to take action to make sure this sort of thing never happens again, while nevertheless protecting our ability to do the kind of science I know you also support.
Well, I think we have made the world a more dangerous place by failing to solve this problem. And by that, I mean, if you're in Pyongyang or Tehran and you want to bring the Western world to its knees, you've just had a demonstration of just how cheap that could be. You wouldn't even need a very dangerous virus, just a highly infectious one.
Okay, I'm sorry, but we are actually literally seeing what's happening in Tehran right now. And it's not the viruses that are cheap, it's the drones. So that is just one of the dumbest things I've ever heard in my life. And I cannot believe that he even just brought that up.
One, and you could destroy the Western economy almost altogether. And therefore, the message, and by the way, the World Health Organization would shrug and say, I'm afraid we weren't able to find out, and we didn't get enough cooperation from the Chinese. That's a pretty clear message.
What does he expect the WHO to do, by the way?
That this is a way to go. And I hate to say this because it sounds like I'm sort of recommending it to them. Of course I'm not. I'm just saying that I think we do need to take that into account as well. By the way, just a little footnote on that. Spare a thought for the people who sell mammals in markets. Nobody really minds too much about throwing them under a bus, saying, well, I think it was you guys who did it. You know,
what?
They've got a livelihood to make too.
Do you see what I mean?
God. So are you kidding me?
The live animal trade should be regulated, completely regulated, and we have totally neglected this as an issue, and we are going to see another pandemic because of the live animal trade, and that is just irresponsible.
It's unbelievably irresponsible too, because first of all, of course, I've spared a thought for all of the people who were working at the Huanan market and involved in the live animal trade.
Sure.
But the criticism that I have for the live animal trade is exactly what you just said, Gigi. It's that it's unregulated, not that we need to end it, because I do spare a thought for those people. I don't think that they should just be out of jobs because Matt Ridley and Jay Bhattacharya are like, animals gross, like animal sales, whatever.
Well, let me just make one more
point. That attitude about wet markets. And the fact is, they're culturally important. There are multiple wet markets in the US. There's multiple wet markets here in New York City. So the idea that this is something unique to China, or that, oh, we're not thinking about the poor animal vendors, is ridiculous. It is an issue of regulation and making evidence-based policy, and that's what we should be doing, and that's what we're not doing.
I just have to add that, you know, there are things that we could have been doing after this this pandemic started. We could have been pressuring China to clean up the live animal markets. We could have had them. We could have pressured them to do more regulation. We could have stopped much more illegal wildlife trade. We could have made them create like parks to have more separation between bats and people. We could have done a lot of things, but we couldn't do that because we soiled the politics, you know, almost immediately. After SARS-1, we did pressure them to take a lot of measures that made their lab safer, that you know, they invested a lot more in public health and surveillance. I mean, after SARS-1, there were a lot of mistakes that the Chinese made, and because of the World Health Organization pressure, they cleaned up their act. And so I think, you know, we just had a missed opportunity.
Well, and except they didn't clean up their act, which is exactly why the Chinese government doesn't want the pandemic to have started with zoonotic spillover.
Sure, but I mean, they did create something like a CDC at that moment.
And they did a lot of stuff. And that's exactly why those memos were written by the State Department, because we were working with the Chinese government to actually help them build up their scientific research infrastructure. But the motivation for China to not have it be from the market, because everybody assumes that the Chinese government wants to cover up a lab leak because they would be blamed for it. I think it actually looks much, much worse for the Chinese government to have this emerge exactly the same way that it did as SARS-1, right across the river from their national marquee coronavirus research institute. I mean, it is hugely embarrassing for the Chinese government, because all of that investment, everything that they did to try to prevent another SARS-1, happened just because the live animal trade was still occurring, and it was still occurring in an unregulated way, which is why initially they denied that there was any live animal trading at all going on at the Huanan market.
Left, since there's two folks over there. And if you have a question, please line up at the provided microphones.
So, Matt, all the issues that you faced with peer review, do you think that an emerging model like the one that we see for the journal eLife, where you have publication and then after publication you have open peer review, do you think that would be a better model for this particular case?
Yes, I do. I think I don't spend much time trying to publish.
How could they have not found a journal? It's not a credible excuse.
On it, but it does seem to me that the problem of PAL review and gatekeeping, in other words, keeping out people.
It's also interesting here, right? Because he's saying the scientists are wrong, the experts are wrong, the media is wrong, the journals don't want our papers. That's not the underlying problem, right? The underlying problem, of course, is.
And I think it largely derives from the asymmetric anonymity.
It's not because of
the reviewer can be anonymous.
They didn't tell a coherent story, and they couldn't tell a coherent story no matter if they had laid it out. So, I mean, that's why they can't release it.
Yeah, I think it's super problematic that it's like, why are they the ultimate authority in this space when this isn't even their area of expertise? Yeah, I don't understand
that. Well, Anna, welcome to the miserable world of our reality.
Don't try and make sense out of it.
Don't try to make sense out of it.
But the author of the paper is not anonymous, then I think that creates a big part of the problem. It does seem to me, in an age when publication can be instant and there's an enormous we're drinking from a fire hose of information on the internet.
I mean, he wrote a whole
anyway,
book.
There's no reason not to allow people to just put things out there and have the peer review happen in open session with people tearing your paper apart in front of the whole world. I think the only people who would suffer from that are those who make a fortune out of publishing these journals that rely on the existing kind of peer review.
Actually, can I ask a follow-on to that question? Because I think that's such an interesting point. So there's been, as you know, and you pointed out in your talk, an enormous fight over that proximal origins paper and whether Nature should or should not retract that paper.
It's Nature medicine.
And let me ask again a question about the stakes, right? Does the editor of Nature get to decide what's true about proximal origins?
Well, strictly speaking, it's Nature medicine in this case, but I don't know whether that comes under Nature. I don't really know.
How do they not know this?
This does seem to me to be
scientific journals.
I mean, we
just clearly never tried to publish there
about how journals are terrible, and they literally don't know the difference between Nature and Nature medicine. Like, Jay is an MD, theoretically. I mean, he does have an MD degree, even though he's never practiced as a physician. They should know what these journals are, and that they are two different journals within the same journal company. Matt just talked about the journal corporate structure, and yet he doesn't understand the difference between these two journals. It's pretty remarkable.
Yeah. And to be clear, they're different journals.
If you walk
they are different journals
down the street in America or Britain or anywhere else, and you ask your friends, what do you think happened? They all say it came out of a lab. Come on.
Yes, because you guys have nonstop been saying that
about it.
The one
It's actually somebody I talked to today was like, "Oh, it came out of a lab, right? That's the most recent information." I'm like, "Where did you hear that?" And they're like, "Western media." And I'm like, "No, that's not." There's multiple camps of thought, and it just and
they're
because of these guys.
It's true. It's because this is how they talk.
And they've been talking about this for the last 6 years.
If they really thought they were wrong, if they really thought the rest of the world was wrong on that, get out there and debate it and so on.
We don't need to, because we publish papers on it. You can publish a counter paper, but you evidently can't get it past peer review. All of us gatekeepers.
It's just not credible that they couldn't find a place to publish it. It's just not credible.
That doesn't feel to me right. There's too much power with these
PLOS One, I hear, is still accepting submissions.
Why I've been so
in the Lyme disease world, they would have just created a new journal. Oh wait, that's what they did. They created a new journal.
I'm not introducing it. This is what all we scientists believe are the standard of scientific truth, right? Independent people looking at the same question come to the same conclusion. That's always been our standard of truth. Whether or not top editors of top journals decide one way or the other is, in a sense, truly beside the point.
Yeah, I mean, scientists, like anyone else, are prone to confirmation bias. They look for evidence that supports their theory and not evidence that
that's a little bit of mirror imaging going on right there.
And you can't expect them not to. That's human nature. But what?
Who was their audience?
The difference between science.
There's not too many people in the audience.
Yeah, like it's like you're speaking to scientists who agree more in this camp that we're talking from. So it's like, who are they trying to convince about their information? Like, I don't really understand that.
Anybody.
So I have 2 questions. What is the epidemiological evidence proving the lab leak theory? And in the name of gold standard science, which we're all dedicated to, can you describe in detail the experiments that you personally performed to support your lab leak theory? Thank you.
I love this woman.
The second
that was great.
Question is very easy to answer, which is that I have performed no experiments at all. I'm not an experiment.
Well, good for him.
Analysis.
The first question: what is the epidemiological evidence for a lab leak? And there is no direct epidemiological evidence, I think, that could distinguish between a lab leak or a zoonotic.
It's like a whole paper you could reference.
Yeah.
Definitively. However, I personally think that the fact that we had a virus extremely well adapted to infectivity in human beings is unusual for one that has jumped.
Yeah, because pandemics don't happen all the time.
Would be expected for one that has spent a lot of time in cell cultures.
In
Ebola
cell cultures.
And SARS.
Well, then how did it get that fear included site? Because that's the first thing to go bye-bye when you stick it in some
beer. Can be very lethal, but not very.
Famously, the 1918 flu pandemic didn't happen because it didn't actually.
On the human species. This one was extremely infectious and fairly mild from the start. That's very unusual, I think, for a zoonotic jump, and therefore that is epidemiological evidence.
Having never studied any of them, I will continue making
the other one piece of evidence that's crucial, of course, is that the phylogeny of the viruses very clearly suggests that it did start in Wuhan and nowhere else. The evidence on that is pretty strong.
Can I do a follow-up to that question also?
Yes, we need to talk about that.
What is the nature of the evidence that would definitively establish it as a lab? Because it seems to me that there is no lab experiment that could, in principle even, distinguish between the two definitively, or even direct epidemiological evidence, short of the kind of epidemiological evidence you get from looking at lab notebooks, for instance, definitively establish.
I think we're unlikely to find an absolutely definitive, dispositive smoking gun inside the genome of this virus. As I said, we found some extremely surprising and suggestive things, but they don't completely answer it. If you found an account of an experiment with a chimeric lineage 4 sarbecovirus that was 98 to 99% similar to this in Wuhan in 2019, then I think we're home and dry. But I don't know whether we will ever find that.
I mean, it seems to me that
they didn't have it.
Key piece then of the case you're making.
You can keep on looking and saying that you need to find it.
Evidence directly, not epidemiological.
Can't prove a
directly,
negative, right? This comes back to one of the
directly.
basic principles of science.
It's the combination of the three.
It's very much a combination of, it's the good old Bayesian thing. You add up probabilities.
He's going to keep making money off of this for years to come.
Feels.
I love the drop.
I mean, a better analogy might be.
So there's a new disease that we can.
You're putting before.
When he moves over to measles.
all the evidence, and the other side is putting their evidence before the jury, and the jury has to make up their mind. And I'm pretty sure that if this was put before a jury, that at the moment the Lab Leak would win the case.
And
hi,
that's the kind of reasoning that really appeals to Jay Bacteria.
Really interesting subject. I'm
Andy,
thinking
can you just
about
pause 1 second?
a related topic in the spirit of the.
Because I just wanted to inject one thing here, right? Because Jay asks a very, reasonable question, which is that what would convince you that came from the lab, right? And we have all answered that question, which is any evidence that it was at the Wuhan Institute of Virology prior to the pandemic, right? But the question he doesn't ask is what would convince you it came from that market? And it's important that you ask both questions, right? Because I am genuinely interested in that. If you can show that the early cases clustering around that market, there's clustering within the market where there's all the animals, we find samples in which you find the animals and the virus in exactly that place where it's clustering and so on and so forth, that's the kind of evidence I would say, like, that's pretty good evidence that it was probably there. If you had replaced that market by the Wuhan Institute of Virology, the early cases clustered around that, and the environmental samples was in the BSL-2 lab, there you go, it's the lab, right? So that's the kind of evidence that you would want to get to point to a lab, is exactly the kind of evidence that we have that points to the market. And to be clear, the market is where we would expect a pandemic for this, like this, to actually happen, is that market in the first place. And Angie, I think you're trying to chime in, but you actually muted. So I don't know if there's something on the audio again.
Yeah, we can't hear you.
The spirit of the academic freedom lecture series, artificial intelligence research is advancing rapidly. There's currently no regulations on deep learning or artificial intelligence research. Despite that opens up a lot of opportunities for harms, dangers that are being warned about by some experts in the field. Synthetic biology is connected to that, whether you had a case of bioterrorists or even a misaligned superintelligence connected to a self-driving lab or something like that. So I wonder if either of you have thought about this subject as something where the scientific community, maybe even NIH in particular, should be thinking about how to approach the dangers of scientific research, or if it could even be the topic for a future academic freedom lecture. Thank you.
That's a great suggestion for a future academic lecture. So we absolutely will do that.
Yeah, I agree, and I will look forward to reading it. And I don't claim to be an expert on that area, except I would say one thing, which is that I'm not sure that the people having the artificial intelligence debate currently have talked to you guys enough, have talked to biomedical scientists and evolutionary biologists.
Are you kidding me?
Because, for example, this idea that, you
clearly has not been in DC long enough to get to enough AI bio lectures.
He did say that he's not an expert in this.
World and kill people if necessary to get to your paperclip manufacturing goal.
I think the next presentation clearly needs to be a mirror light.
AGI could take over the world.
I hope that it's actually about
that all the time. They're called
autonomous
viruses,
paperclip factory that like starts building Terminators or something.
Do damage to us, but they have competition. They have competition from other species, and the same will be true of artificial general intelligence. There will be competing AGIs in an evolutionary ecosystem.
Okay, he really does not know what he's talking about.
Yeah, I don't.
In this topic as well as the first.
I want Christian to do an analysis of how ChatGPT is evolving relative to Claude.
Thanks.
Oh my God, I know they're going to have Jamie Metzel on for the next.
I want a tree.
My point, yes, is that we need to understand that we're scientists. That's what we do. Actually, so if we give up on that, we better do something else. I think that it's in the nature of what we do. But that same need to understand actually is what's driving my question to you, because we have to make decisions of what we fund or not. And so there's always this argument and counter argument. In the gain of function, the counter argument of why you would want to actually promote research is if you find yourself in a situation with a pandemic or an epidemic, you want to be able to provide the tools to counteract it, which was the argument that has been put forward. So how do we, those that are providing resources to others, judge these elements? I mean, to what extent any type of knowledge, if taken outside of the context, can have malevolent applications? And so, but we have a responsibility of thinking about it. It's not that because it's an inevitability, we're not going to do it. So I'm curious to see what you're thinking in terms of course, we've created these committees that evaluate and determine is this appropriate or not, but that requires cooperation. And are we currently in a state where we are going to be able to get that cooperation, particularly where science has no boundaries?
Good question.
I'm going to go off a slight tangent in answering this, and I might not give you a very good answer, therefore. And that is to say that I do think there's a possible lesson that we need to be trying to learn here, that lab-leaked pandemics might look different from natural pandemics in terms of particularly how infectious they are from the start, and therefore whether things like lockdowns would work. In other words, if it happens again that there is an outbreak somewhere in the world, one of the questions to ask really early is, has this been in human cells on the lab bench for a few months? Because if so, we're going to have a real trouble confining it and cutting it off and using social isolation and NPIs.
This is the dumbest thing I've ever heard.
If it's literally just come out of a pig that
a lot of competition, Angie,
dropped by a fruit bat the week before, probably we're going to be much more successful with isolation and that kind of tactic. So the public health
again, 1918 flu,
managing an outbreak.
And 1918 flu, I should just add, at least according to some of our co-authors' work, yet again, that Christian and I didn't contribute to, but that they've done on the origins of 1918, show that it actually spilled over relatively quickly from birds before it went into humans. So everything he just said is completely disproven by 1918. And we know that not only from epi data from 1918, but we actually also know it when the acting NIH director, Jeffrey Taubenberger, reconstructed 1918 in probably one of the most flagrant gain-of-function experiments ever and started messing around with that virus. Those experiments, I should add, were very, very important for us to understand how flu pandemics occur and how they cause disease and how they kill people. So I'm not trashing that work. That work was very critically important for our understanding of pandemics. But it also completely disproves everything that this man was just saying. And for coronaviruses and SARS coronavirus 2 in particular, when we grow that virus in viro cells in the lab, or even sometimes in some primary human cells, it loses the furin cleavage site and it acquires other cell culture adaptive mutations that actually make it a less fit virus for growing in vivo in any kind of host. So everything he just said is completely stupid. Unsurprising and pretty consistent with many of the other things he said. But this is one of my particular things that I find to be incredibly stupid, just because it completely misunderstands how any viruses work, and it completely misunderstands the different evolutionary steps that viruses have to go through when they are actually switching hosts and turning into a virus that will be efficiently adapted to a new host.
Ought to be taking into account the mode of origin, not in a black and white way, but as a possible influence. That probably goes well beyond my expertise, and I might well get shot down by someone here, possibly by him.
Oh, really?
I will not never do that. Although maybe we can have a discussion about lockdowns at some point. Actually, I wanted to bring that question, a fantastic question, to sort of lift it up a little bit more, and ask what responsibility do scientists have, right? So like the story I love to hear to tell about the launch of the nuclear age. Enrico Fermi does this experiment on a squash court in the University of Chicago, of all places. He starts a nuclear chain, the world's first nuclear chain reaction started by a human, on a squash court, literally in the middle of Chicago. And before he does this, thank God he's a great mathematician, he does a calculation of the likelihood that the chain reaction can be contained, and decides that it can be. And then he does the experiment, a socially responsible thing to do before he launches the nuclear age. Shouldn't we scientists always do that calculation?
We do, Jay.
And in Los Alamos, they did take seriously the possibility that the first test of a nuclear explosion would ignite the nitrogen in the atmosphere. And they did the calculations and showed that it would not before they did it, and rightly so. So yeah, there needs to be precautionary calculation going on.
We do take precautions, extensive precautions.
They're not interested. Actually, they're not actually focused on any biosafety.
No, I know that they're not.
Intelligence agencies. As you said, we know that a number of intelligence agencies have looked closely at this. Like you, I would love that research and conclusions and evidence to reach the public someday. I hope it will. But I think maybe you could make a small argument from what we know about the intelligence conclusions against the lab leak theory. So for example, you mentioned the 3 sick researchers. There's these allegations that 3 researchers at Wuhan Institute of Virology who were very involved in these experiments got sick in, I think, November 2019. And if true, this is evidence, some possible evidence.
This is November,
at least partially in favor of a lab leak.
Well, he talked about
But
September,
if you were an intelligence agency, you had rock solid evidence that had happened, that those identities were correct and confirmed, that they were severely ill and so on. Wouldn't that make you almost certain in the lab leak scenario? And we know that the US intelligence agencies have been pretty ambiguous about this. Most of them reached only a tentative conclusion. If I remember correctly, the FBI had only moderate confidence in a lab leak. So doesn't that suggest that maybe they didn't have such strong evidence after all?
Yes, I think you're absolutely right that if they were absolutely certain they had COVID, then of course they would be 100% certain that it was that. But people get colds.
Just to be clear, the conclusion is actually in the intelligence report that they did not have COVID.
Yes. Exactly.
So it's not just that they don't think it was COVID in the first place, right?
And why would you actually ask the people what virus they had? Granted, Ben Hu is an excellent coronavirologist. I like to think that I am too, but I couldn't tell you if I got infected with, 229E or OC43 or rhinovirus 1A or whatever. I can just tell that I have a cold. I mean, I don't even think that this met that bar of I have a cold. It was just some shit that Mike Pompeo made up.
Yeah, that's the thing. Like the whole thing about 3 infected workers at the Wuhan Institute of Virology in November, right? First of all, if there were actually 3 young individuals infected at the Wuhan Institute of Virology in November, there would have had to be a much, much, much bigger outbreak ongoing at the Wuhan Institute of Virology at the time, for which we have absolutely no evidence. And in fact, there was a Western researcher, Daniela Anderson, who worked at the Wuhan Institute of Virology at the time, and she was asked this question: were there any infections at the time of this supposed November dates? And her answer was no. So, and again, the intelligence community concludes that these are very likely not COVID-19 cases.
And a lot of times, I'll just add this. I don't know what the practices are at the Wuhan Institute of Virology, and perhaps I'm a little more generous than some of our colleagues here at the NIH in assuming that they have similar practices to many of ours. But if we have any kind of respiratory symptoms or any symptoms that are associated with the viruses that we work with in the lab, we are instantly reporting that. And if you don't report that, then, and you're in my lab, you are running the risk of not being in my lab anymore, because I report if I am traveling and I'm in another country and I haven't been in the lab in weeks, I will report respiratory symptoms, because better to report than to not. And that is what most labs do. That is their practice. That is one way that we carry out what's called medical surveillance and make sure that we aren't getting lab acquired infections. But if you work with a respiratory virus, any respiratory virus, and you develop respiratory symptoms, the most containment facilities, you will be required to report that. If you show up at work and try to hide it, you're out. And it just doesn't make any sense at all that if people did report that they had respiratory symptoms when they're working with respiratory viruses, that it wasn't followed up upon, especially given either September or November timing.
Just what they had, they just think it's intriguing, presumably, that, I mean, they say they were sick enough to be treated in hospital. Ben Hu
knows that everybody has to go to the hospital if they.
China, this is the way in which you see your GP.
The problem here is that I'm not party to the evidence. I don't know how they got it. They don't want to.
You're not party to anything
because that would presumably compromise.
Now it's the time to play dumb. Just asking questions.
Like that.
I'm just asking questions.
So
I just got intrigued by this at the beginning of the pandemic after I learned that Christian was a liar.
Which is why it was just one loose end that
I believe this paper at first,
but
then I just had so many
I don't
questions.
myself find it to be part of a strong part of my argument.
Oh, it's a strong part of my completely uninformed argument.
Can I follow up on that question? By the way, this spirit of like pushing back is fantastic. This is what this this
vigorous great work. Great work, gold star, gold
so
standard.
Let me follow up with that question on the nature of our cooperation with states that hide evidence around this. Right. For the longest time, the West has cooperated extensively with Chinese scientists, and it was extensive and very fruitful collaboration. Let's go back to the Cold War. The Cold War, one of the major issues in the Cold War was that scientific collaboration. And in fact, there were two scientific worlds all throughout the Cold War: the Soviet bloc and the Western bloc. In part because the thought was that there was no way to do fruitful collaboration that wouldn't result in exploitation of scientific knowledge developed in the West. Where are we on that, in your view, given the evidence that you've shown and your long thought on this issue? Because it seems to me international collaboration is a very, very important, fruitful tool for the advance of science. And yet, if it's not possible to have that collaboration without a worry that on crucial scientific questions like this one, you're not going to be able to get collaboration from the Chinese authorities, what should be the right policy? I mean, I can tell you that we're still thinking about that.
Are we almost at the end here?
Decision on that. Where's the UK on that, and how are you thinking about that?
Well, I think that question is well above my pay grade, actually, but.
Does he have a pay grade? He's literally inherited like a manner that looks like Skyfall from like that James Bond movie.
On the one hand, I would like to see politicians and all others who are collaborating with China make this part of the conversation. I mean, we do that for human rights. Why don't we do this for this issue too? Look, saying at every meeting, yes, we would love to collaborate with you on something. Please, could you be a little more cooperative in sharing information about what happened in Wuhan in 2019.
The idea that I would say this to any of my Chinese colleagues.
And I see no evidence that is happening, even though there are many, many institutions in America.
The idea when we had adults in charge managed to have fruitful scientific collaboration with the Soviet Union and with China.
Dealing with, say, I don't know, Indonesia or Brazil.
A lot more about what was going on in both those places because we had fruitful scientific collaboration.
They opened the books.
These people are lightweights. They don't know anything
of that collaboration with
we are much too frightened to.
My husband is older than me. He moved to the U.S. for his postdoc, and it was only because of Glasnost and Perestroika that allowed those collaborations, which allowed him to make the relationships that got him his postdoc in the U.S. and allowed him to move to this country. Live the American dream.
Don't include this.
Well, the American dream,
because I think it's.
The U.S. cannot close itself off from the rest of the world when it comes to science, and if they try, then they will no longer have science.
And we're about to see
you brought
what
up
that looks like.
Let's just do this.
My understanding is that there's plenty of evidence to suggest that it naturally occurs very often, particularly between the S1, S2 junction, like you were talking about, in many coronaviruses, naturally occurring coronaviruses, and in many other viruses. that infect humans. And actually, there's an evolutionary advantage to having those furin cleavage sites. So you brought that up as a smoking gun. Can you explain that, given that it's naturally occurring, why it's still a smoking gun?
Yes, furin cleavage sites naturally occur in MERS-like viruses, Merbeker viruses, most of them, in many other coronaviruses, many other kinds of viruses, never in Sarbeker viruses. That's the point.
No, we've never sampled one.
We have one virus that has it.
People say, oh, you don't. Of course you get furin cleavage sites in coronaviruses. Yes, but we've never seen them in Sarbeker viruses before.
And I should add that one of those banal viruses is one amino acid.
Remember I said that this virus's natural habitat is the guts of Rhinolophus horseshoe bats, not the lungs, the guts. It's an enteric virus in bats. And in those tissues, and using the receptors it does in there, cleavage is a disadvantage. Cleavage actually makes the virus less able to invade the cell.
Furin cleavage.
That's the hypothesis. I'm not saying that's a proven fact.
It still gets cleaved.
So furin cleavage sites
it's true though that, like, again,
against
the main difference here is that, you know, bat coronaviruses in general, while there are, in fact,
that's the argument as to why we've never seen,
as we're looking at, for example, in rodents,
it's hard to put a number on how many we've looked.
I was just gonna, I wasn't sure if you're gonna keep talking, Christian, but I'll just very quickly say here too, that, I mean, this is one of the things that drives me crazy, right? Because I work on the host response to infection. This is a very simple thing. You're not going to get a lot of furin cleavage sites coming out of bats because they don't express very much furin. So it's not there to create a selection pressure that would lead to them evolving a furin cleavage site for natural reasons, because furin is a convenient protease to use in the bat gut. They're going to use other proteases because there's a shitload of other proteases in there, including those cathepsins and things that I mentioned earlier, and it still gets cleaved. He is making the argument that in bats, the spike protein does not need to be cleaved at the S1, S2 junction. There is no such thing, at least as far as I'm aware, as a host that doesn't require this cleavage to occur for the virus to complete its life cycle. So once again, this is getting way into the weeds about the virology, but the host is very important for evolution and for some of these features emerging. But it's not because, no, furin is terrible and it's bad for the virus if it's in bats. It's just because the bats don't express very much furin in their guts. So the virus has no evolutionary incentive to evolve a furin cleavage site.
No, and to be clear, Andy, it might be selected against, right? But one key thing about this is that the SARS-CoV-2 furin cleavage site is really suboptimal, right? So first of all, that's the opposite of what a researcher would do, including the papers that Matt himself linked to, where they're optimal, perfect furin cleavage sites.
I don't know, Christian, I might want to make a challenge for myself. Do a shitty furin cleavage site and see how well it works.
See how well it works. But so this one is suboptimal, right? And I will say, like, as we are looking, so again, in the Sarbeker, like, we have only sampled a tiny subset of all the ones that are actually out there. And as you know, these are standing bets that we have. Are we going to see, you know, bat furin, like Sarbeker viruses in bats that have furin cleavage sites? And I actually think as we're looking more, I do think we will. They may well be suboptimal, right? But it is true that we have not found one for Matt's like point that we have never found a virus with a furin cleavage site. And Sarbeker viruses is not correct, right? Because we have SARS-CoV-2.
We have SARS-CoV-2. We have Banal 52, I think, is the one with the almost furin.
No, I think there are other ones. Like, I think you're referring to the ones from Christian Drosten. Those are actually European viruses, but directly from bats. I don't think they've been Banal viruses. I don't believe have like an insertion there. But I will say, though, that there are other viruses, and this is to the point of the person who asked the question too, is that this S1, S2 region is probably the most evolvable part of the entire coronavirus genome as we're looking across not just the Sarbeker viruses, but beta coronaviruses in general, is that this is the most evolvable part of the genome. And we see insertions come and go there all the time as we're looking across the broader subgenus of Sarbeker viruses, right? So again, the fact that a furin cleavage site might be an insertion that you might see, and then as soon as it finds itself in a respiratory environment, It's going to become fixed at that point, right?
Yeah.
So again, it's not like an unusual thing at all. The fear and cleavage site is also quote unquote out of frame, which is something a designer would never do, right? But it's something that nature does all the time.
Except for Matt's co-author, who once said that she is an experienced cloner, frequently clones things out of frame, but it doesn't really matter because it still works. I guess, like, yeah. It just happens all the time to us experienced cloners, although I can't recall something that just accidentally went in out of frame that worked also.
It's just not the way in which it's done.
Yeah, at least that hasn't been my cloning experience, but hey. Anyways, we'll finish this up. I think we are very close to being done. Thank God.
We've looked at, and the proximal origin paper said, don't worry, we're going to find one in a sarbecovirus soon. Well,
me to it.
Full time. One. But it's something like 800 different strains of sarbecovirus.
Haven't changed your mind on that one.
One with a natural fear in cleavage
site
in it.
Not correct.
Next question, please.
Yeah.
Thank you for a tremendously well-organized and compelling narrative.
Oh.
One of the things that was most disheartening to me during the pandemic itself was the scientific community's embrace of the conspiracy theory jargon that went on. And I'm wondering if you can talk a little bit about the sociology of science and the responsibility of scientists to combat
I'm curious.
This whole idea
the political appointee.
That a perfectly valid
red flag looks like it.
Alternative hypothesis is a conspiracy theory. We're also fighting against this kind of disinformation language that's being very commonly used to squelch alternative ideas.
This is like the most Orwellian question that's been asked so far.
It's got to be a political person.
Yeah, he is, alleging a conspiracy between the former director of the NIH, the former director of NIH, the former director of the Wellcome Trust, and a bunch of international scientists that have published in journals, right? I mean, that is about as conspiratorial as it possibly can be when you elect the intelligence communities, the journals, the media, like everybody is in on what he talks about. There is a conspiracy to suppress this particular one. I mean, it is bog standard conspiratorial thinking and conspiracy theories, no different than a lot of other conspiracy theories out there.
Yeah, and I mean, it's the same right down to the gaslighting in response to it, where it's like, you can't call us a conspiracy theorist. That's so rude. Like, I don't like the use of this conspiracy language. It's so unprofessional and insulting, and this disinformation language. Okay, well, I won't call you conspiracy theorists anymore. I'll just call you fascists who are trying to undermine democracy with your conspiracy theory.
I mean, I do think, like, because, like, again, he's going to mix up, like, everybody who believes in a lab leak is called a conspiracy theorist, right? And that is not correct.
That's
right? Again,
correct.
The idea that this could have come from a lab is a very valid scientific theory, especially given the proximity to Wuhan Institute of Virology, right? So, like, that is a perfectly valid and necessary scientific hypothesis that must be taken seriously. But again, that is not the same as to say, like, a lot of the narratives that are then presented around a potential lab leak, which is what Matt gave a good example of today, right, are indeed conspiratorial in the framing, because they say, like, don't trust any of the data, don't trust any of the scientists, don't trust them, don't trust the journals, don't trust any of them, because they're all in a conspiracy.
Yes, don't trust the experts.
Yeah. Exactly. Don't trust the experts.
Well,
right?
then
that is what we're talking about. Yeah, go ahead, Anna.
No, just like, who are you supposed to trust? If you can't trust everyone,
that is the only people that you're supposed to trust.
Yeah.
And that's what they're setting this up to be, right? Is that they are the ultimate authority on all scientific topics, even if they're not experts in this particular area. I'm not an expert in virology. I'm actually a program specialist, not a program officer. Like, I'll be completely honest about my credential. Like, I don't know certain, but that don't claim to. And it's just the way that they say, oh, well, I'm not an expert, but I actually have all of these opinions that lead me to present myself as an expert is very problematic and concerning.
Yeah, you nailed it, Anna. I mean, that's exactly what the situation is. And essentially, they are spreading conspiracy theories, as Christian said. I mean, these are tactics that have been well developed and they work. And that's why they're using them. But the way to respond to them is to turn around and complain that we don't debate them. We don't talk to them. We don't take them seriously. They've been complaining about this for 6 years. I mean, if I had said, let's have a thing and let's invite Matt Ridley to it, I actually bet he'd probably come, because I don't think he'd miss the opportunity to start interrogating Christian. The problem is he probably wouldn't come if it was something that we were running, because he wouldn't be able to completely stick his finger on the scale and make it look like we're all liars and you shouldn't trust us and you can only trust them because we've been engaged in this big conspiracy theory to hide the truth from you. We're all getting paid by Tony Fauci, Christian foremost among us. And, we've got all these incentives. I mean, the fact is, I get called, a pharma shill all the time. I have not made a dime from pharma. Like, I haven't even gotten a grant from pharma. Like, they want me to help them with all this stuff, and I'm doing it for free. Like, it's just communication and stuff, and I'm pro-vaccine. Like, so great, fine. But that doesn't mean that Moderna is paying me to tweet about mRNA vaccines. They constantly say that kind of stuff too. I mean, they just constantly throw this stuff at us. And then they say, you're not having a reasonable discourse with us. You're not respecting us because you're using the D word, disinformation. And you're saying that, we're conspiracy theorists. How are we even supposed to talk to you when we're so rude like that? But the fact is, they are conspiracy theorists. They're talking about a conspiracy that's unproven, and so it's a theory. It's actually a conspiracy hypothesis.
Yeah, and just to be clear, because he is very specifically talking about me, right?
Yeah,
and he's talking about how I didn't actually really believe what I was saying, right? Like, I know the facts of all of this, right? And again, I know what he is saying is just false, right? And he is then alleging that there is this major conspiracy, which I know there is not. Right. Again, it is the bog standard definition of what a conspiracy theory is. But really importantly, again, though, it does not mean that anybody who believes it came from the lab is a conspiracy theorist. Right. Correct. It is not the same thing. Again, the belief, the hypothesis is perfectly valid. Right. It's just the evidence that we have very clearly shows that actually it wasn't a lab leak.
I should probably rephrase, Christian. When we looked at this, when you looked at it with proximal origin, and then when we all got together as our larger group of Zooinati and started looking at it, we treated it like a hypothesis that we were testing.
Oh, for sure.
When they talk about their conspiracy theories, they aren't talking about testable hypotheses. They're talking about Matt Ridley is more of an expert on what went on in your head than you are. And I'm quite certain that Matt Ridley is not clairvoyant. If he were, he would probably change his personal style because he'd be able to read what I think about it. And it's pretty scathing, but I'll save that for another live stream. Let's finish this trash and then have a closeout discussion.
And have done for many years. I've been a science journalist ever since I joined The Economist as science correspondent in the mid-1980s. And this question has always been there. How do you take mavericks seriously enough to not miss the genius? And how do you not do so in such a way that you end up listening to cranks? And it's very, I know no rules of thumb that enable me to say, this guy's a crank, that guy's a maverick genius. Yes, in some cases, it's very obvious, which side of the line people fall on. But I do find it hard to be absolutely sure in certain gray areas. And I want to remain open-minded, but not so open-minded that my brains fall out, as someone once put it.
He used that line a lot.
So in this case, I took the view that it was unlikely that it came out of a lab because the people who knew more than I did about virology were saying so. And I also, my prior was, we're not that good at designing viruses yet. Well, I was out of date. We are that good. We are able to swap spike genes into viruses very precisely and create viruses with much higher.
Yeah, I mean that is basic virology.
So I was wrong on
decades and decades.
But when in 2020, when Facebook literally censored anyone mentioning a lab leak, and so did Reddit, but Twitter didn't.
I'm so sorry for the people that are there.
That is not
It
true.
downplayed it a lot. It was very hard to
This is
have
the
a conversation
we are being
as a
silenced
journalist
part.
or writer or
I mean,
politician.
Yet here he is giving a talk in front of the NIH.
This.
What I love it is that
and
his co-author literally, and he got a book deal by tweeting about this.
By people
and
like the New York
an
Times.
article was
Now,
literally written about his
even when I
co-author
thought
Alina
it wasn't
Chan
right,
that said how
that
Alina Chan tweeted the lab leak
I
into
knew
the
that was
public
going too far.
consciousness.
It wasn't a conspiracy theory. It's a logical possibility. There've been lab leaks before. So this feels to me like one that wasn't difficult.
Except like several of the examples he gave were not actually lab leaks, but yes.
Yeah, and plus those are lab, like
Let's
again,
look
the
at
big,
it.
we need to
might
talk
be
about because
against it.
the big
We
difference is that
mustn't
those are known
label
viruses,
it
right?
in this
Already
cheap
existing
and,
viruses, selected because they
really misleading way.
Thank you for that question.
Except for the weaponized anthrax that the Soviet Union was
Thank
making.
you for
but
putting this on.
They knew what that was
I'm
too.
here to be open-minded, but some people have laughed at the notion of talking about this, scientists and people in the general public, and I just wanted to know what you thought about that, and what could be done about that, perhaps, for scientists and the general public.
So let me take that question. So the question, I guess, is there are people in the general public and also scientists who think that maybe this question ought not to be discussed, especially on a stage like this at the temple of all science, the NIH, right? I mean, I should say I just strongly
reject the high priest,
right? I think that if we can't ask and have reasoned conversations on this topic, that is I believe at the heart of a lot of the public distrust in science, that the future of public support for science is very dim.
Yeah, but we wouldn't have that problem with trust if you weren't up here lying about
wherever
this
they
stuff
lead,
all the time.
if we are to
Yeah,
public trust in science.
I fear for the future of science too, with you in charge of it.
I live in Britain. Britain is a leading biomedical nation, probably second only to America and China now. You know, but you know, it's got a terrific track record.
It does, but
and I'm a fellow of the Academy of Medical Sciences, and I'm not a fellow of the Royal Society. But I wrote to both the Royal Society and the Academy of Medical Sciences saying, please hold a debate about the origin of this virus that is killing millions of people. And the reply I got from the Royal Society said, no, we only have debates about scientific issues.
Yeah.
And the reply I got from the Academy of Medical Sciences said, it's too controversial an issue. Well, I thought the whole point of debates was
just to be clear, he and his co-author gave testimony
the argument I made to them was
the UK
Britain doesn't seem to
about the lab leak. I mean, it's not that they were they were silenced on this, right? And I just one point here, right, is that again, I thought it was a lab leak, right? I brought it up with my colleagues, who then brought it up with Jeremy Farrar, who then brought it up with Dr. Fauci, and then I talked to Anthony Fauci, and let me tell you, he took me very seriously, right? He did. You can see that in the emails, right? He talks about how we have to contact the CIA and the FBI. Nobody dismissed me or anybody else who suggested that this could have come from the lab, right? The only thing that was done there was saying, like, we must take this seriously, and everybody agreed, literally at the highest levels of US science, right? So the idea that that was dismissed and it was not allowed to be talked about is just false, right? And the paper record is there to prove it. You saw the email. Matt even showed the email where Dr. Fauci says we must contact the CIA and FBI, right? So of course they took it seriously, and they still take it seriously, because even though the evidence is very strong to show that this was not a lab leak, it is not yet proven, right? Because it is very difficult to prove something in science. So of course people still take it seriously. It's just this kind of arguments for why somebody like Matt believes that it must have been a lab leak, right, does not hold up to scrutiny. It falls apart as soon as you start digging just a little bit. And that's, of course, what's being pushed back at. It's not the idea itself.
Yeah. And that is exactly how this tactic is used. The second that you push back, they say that you're actually pushing against something other than what you're, or trying to silence them. It becomes another thing. It's one deflection after another. And that's really, we've been dealing with moving goalposts for the past 6 years. Christian and I and our other colleagues and co-authors, and Gigi, I know when you jumped into this more on the policy and like the government and the international relations side of things, I mean, you got the same sort of reaction where it's like, but what about like this other thing that I just made up? And I mean, that's what every single counter argument, or when you try to counter it, comes back to, yes, but what about this other thing that's not even relevant to it?
One of my favorite things is that they rewrite the history of other things that I happen to know quite well. So it's like, it's, the 1977 influenza, Sverdlovsk, like they just are rewriting the history to suit their needs, and things did not happen the way that they said. But I really want to I hope that we're getting so close to the end.
We are less than 10 minutes left on the video, and I suspect that there will be a few minutes of blank space at the end.
Not
so
got
I
the
think
problem
we're less than
which
5
America
minutes
has got, that it seems to have funded some of this stuff. And well, through the Wellcome Trust, maybe that's not quite true, but and therefore we could, here's an opportunity, a sort of geopolitical opportunity to be an honest broker and say, we're going to put a lot of money into this, and we're going to hold proper debates, and we're going to really try and get to the bottom of this. But it seems to me extraordinary that people didn't want it discussed.
Next question, please.
Thank you. I had two questions. The first is, what's your theory on how the virus got from the lab to the wet market? Because to play a little devil's advocate, I appreciate the coincidental nature of it being next to the market, but it's also coincidental that it happened or the hotspot was the market. So I guess, how do you comport the two, and if you had a theory on that? And then the second question, I was intrigued by one of the examples that you gave of a lab leak, I think in Russia, involving anthrax. And you mentioned that it took 12 years for us to realize it was a lab leak. How did we realize that? So,
oh, that's a good story.
In terms of how it got to the market from the lab, if that's what happened, I obviously don't know, and I'm not going to speculate on exactly who walked into the market when they did. But I think it's important to realize that quite a lot of the early cases had not been to the market. So it's the market could have amplified the epidemic anyway and still been nowhere near the very beginning. It could have been a month before it reached.
Those are correct.
Yep, I'm pausing it.
Very remarkable
in
place to
anticipation.
amplify it. Yeah.
I mean, this is just absolutely so completely wrong that there is no evidence of any infections, any human infections prior to the market. And what he just brought up, those cases that were not linked to the market, that is how we know that ascertainment bias, which he complained about at length at the beginning of his talk, is not a factor. Because when we looked at just those cases that had no epi link to the market whatsoever, and Christian, correct me if I'm wrong, because you know that this is not my department. However, the statistical linkage was stronger when you looked at just those unlinked cases than when you included the ones that did have a known link to the market.
That's correct.
They were clustered around the market.
And they were clustered around the market. So what that suggests is that people who weren't going to the market at all for any reason, Wuhan's a huge city. It's 11 million people. It's very large geographically. They were just living around the market. They were doing stuff in the neighborhood, and they happened to run into people from their neighborhood who probably were at the market. And that's how they got SARS too. And that's actually how pandemics start, because they occur in densely populated urban environments like the Wuhan market in the middle of a huge city. That's the significance of the market, because there were the animals there. And that's why we think that this happened.
And it was not just right next to it.
Yeah, what he just said actually takes out much of his early argument that, oh, it's ascertainment bias, and so you have to ignore these papers. He just cited the reason that it's not ascertainment bias, which he either willfully or just because he's ignorant doesn't understand.
Yeah, actually, because again, he makes the argument that, oh, actually the clustering around the market is incidental, right? It's because of ascertainment. But then in the next breath, he says that, oh, but the market is a super spreader event, right? In which case, then it's not incidental, right? Then he's admitting that actually the signal is correct. It's not due to ascertainment, but now it's due to that being a super spreader, right? And the thing is, these are mutually exclusive hypotheses about why you were seeing clustering around the market, right? And you cannot have both. But he just says one in one breath and one in the other breath, right? And you might miss that there can't actually both be true. And again, as I mentioned, it's not ascertainment, right? We know this for a fact.
But Christian,
also, the market is not next to the lab. It's not right next door. It's a half-hour at least drive from one to the other.
And it's across on the other side of the river.
Yeah, it's across the other side of the river, about 20 kilometers away, right? So it is far away from the market.
In a city that size, especially. And then besides that, it's they don't have to build a coherent story, and that's the message here. They can just throw doubt wherever and sprinkle a little bit of reasonable suspicion, because they have not written this out as a full paper where you could poke holes in it like that. That's all they need to do. I mean, that is probably the real reason they haven't published it, because they don't have a coherent story.
I mean, they have written papers, but they are not coherent, at least the preprints that I have read. We've gone over them at length, some of them anyways. And yeah,
I have more to say about Sverdlovsk, but let's hear what he says.
Yeah.
To the market, but the market was still somewhere where it got noticed for the first time, and it took off. A market like that, an undercover market with not great ventilation, huge numbers of people.
Not undercover.
Okay, this market was not secret. It's the size of like a football field, and it's 2 stories, and there's 2 sides to it, and there's literally streets that run through it, and its own sewer system. So
it was pictures that you can see online. It's ridiculous.
It's huge. There's still glasses shops open upstairs.
And just to be clear, he does mention here, like, it's where it got noticed. It's not actually correct, right? Because the way in which we realized, or they realized there was an outbreak here, is because of the level of the hospitals. And they have noticed that they had several patients with unknown pneumonias in late December 2019, right? And then the hospitals realized that actually these are linking back to the Huanan seafood market. So that is how the connection was being made. It's not because it was first noticed there. It's that the hospital noticed that the first patients that we now know today are COVID-19 patients link back to that market itself.
Exactly.
Coming in and out every day is an excellent super spreader opportunity for a virus, is all one needs to know, in a way. So that it's not mysterious that it really took off in the market.
And that is not correct, right?
That's
Again, we have
to
calculate the doubling rate from within the market, and it's about 3 to 4 days, like it is outside the market.
I'm not saying that it had to be a bunch of laboratory white mice that were taken down to the market to be sold as pets or something like that, and I don't want to go into speculative theories of that kind. As for the
okay, so I just have to butt in really fast. One of the lab leak theories is that somebody at the Wuhan Institute of Virology was making like some side money by selling used transgenic mice, like Balb-C mice, which is, I think, is what he's referring to with white mice, but they're albinos. But transgenic mice, selling them to the Huanan seafood market, and that people would be buying lab mice. Now, I don't know if you guys have ever been to wet markets or live animal markets. I mean, I've been to some and seen some where they're selling rodents, including the bamboo rats that they sold at the Huanan seafood market. As somebody who has seen many Balb-C mice in my lifetime and handled many of them, boy, are these mice a lot different than a lab mouse. They are a different species. They are different types of rodents, and there are no lab mice being sold at any of these live wet markets. So it's completely ridiculous. But I actually kind of love Matt Ridley's take on this, which is that they were selling them as pets instead of food. Because, when we infect a mouse with a virus, right? Like we do a necropsy on it usually, and those mice don't actually leave the lab ever alive. And they usually leave either in a tissue digester or an autoclave. But I do kind of like the idea that, oh, well, you're just done with your little MERS experiment here, so I'm just going to take you down to the wet market and sell you as a pet to somebody's kid. It's pretty incredible.
That kind. As for the anthrax outbreak, it's a really interesting story, actually, because Western suspicions that there had been anthrax deaths in Sverdlovsk 66 of them, I think, were presumably coming from intelligence, were aired, and the Soviets said, no, it's food poisoning. And they said, if you like, send some scientists over and we'll show them around. And an international group of scientists went to Sverdlovsk and did a tour of the city and came away saying, no, the Russians are right, this was probably just food poisoning. And it was 12 years later when the wall came down and the Soviet Union collapsed, and scientists came to the West and said, you know what, we left the cap off a filter, or we left the filter off a pipe one day, we sent a plume of anthrax over a suburb, and this is what happened. And so without the collapse of the Soviet Union, we wouldn't probably know to this day what had happened there.
Oh
and
my God.
That's quite an interesting point.
Okay, so one of those U.S. scientists was a spy, maybe. But I'm just going to let Gigi run with this.
I tried to say hi earlier, but you were backstage. Anyway, it was a great lecture, and I applaud the NIH for having this discussion. If we sort of pull back a little bit, there's clearly, you've made an argument that politics was involved in making decisions about what should and shouldn't be done as science. And the implication is that somehow we could extract politics from science and then we would have a better, purer science. And I mean, one would be naive to say that. We need public dollars to support science, and those public dollars come from the public who elect politicians who give us money to do science. So politics is in science. And we're seeing it in this country, you know, very prominently play out right now. And so I'm sort of appealing to you as a visitor looking at this country and seeing how we do this business for guidance and advice, because the NIH is wrestling with us. And, you know, in the spirit of pushing back, you know, I work at the NIH, I see the influence of politics on my own laboratory here every day in a way that is dramatically different than it was, you know, 4 years ago or 5 years ago. And I think a lot of that is the consequence of a kind of mismatch between what science is and what the public understands. And it's out of that mismatch that you had all of these scientists make sort of essentially false statements. But it's because they knew that they couldn't work without contending with the fact that politics is in science. So, you know, I buy all the story. Lab leak, fine. Shouldn't have lied about it, fine. We still can't get rid of politics in science unless you can tell me how.
No, Bevel, you're absolutely right. And of course, politics will always be there, and it's always going to be a part of the story. That doesn't make it right. And that doesn't, I think, excuse going a long way into being political in a situation where there are extremely high stakes. That's what feels to me different about this. Or if you like, just to put it another way, yes, there's politics in science, doesn't mean I can't look for the truth as well. But thank you for
well, to be clear,
I don't think anybody ever said he couldn't.
For your participation and for your engagement. Please welcome, join me in congratulating Matt Ridley for this amazing
congratulations, Matt. Oh, okay, thank God. Oof. Okay, all right, I'm getting this off of the screen right now.
Okay, I have to talk about Sverdlovsk.
Yes.
Can I jump in?
Jump in, Gigi.
Okay, so Sverdlovsk was actually very interesting what happened then. It was in 1979, and there were a bunch of people that died of anthrax. In contrast to the way that he just told that story, U.S. intelligence knew almost immediately what had happened. There were many, many eyes on what was going on in the Soviet Union. We have declassified intelligence showing which generals arrived at what time, what kinds of resources there were, that there was an explosion of some sort at the laboratory that they knew was an anthrax manufacturing facility, at a clandestine manufacturing facility at that time. They, all of this classified intelligence was shared with a scientist, Matt Meselson, who's still around, still on the faculty of Harvard. And he, even though he had this top secret clearance, didn't think that it was sufficient enough evidence to say that it was an anthrax, like an event that was not something natural. He bought the Russian story. It's complicated. There's a lot more to the story, but the declassified intelligence shows that they had him dead to rights. There was never a time that the US government said that it was anything other than what it was. I mean, they had Russian soldiers who were spraying down buildings because there was so much anthrax that was piled into the sky. And so, they were shooting dogs in the streets because they didn't want the anthrax to go elsewhere. So like all of this is very well documented. And so Matt Meselson, I'm not quite sure why he had doubts. Best to ask him. But in at least other people have not had those doubts.
Yeah, and I think, it's one of these things where, Matt Ridley elided the point that the reason that the Soviet Union didn't want that one to be found was that they were violating the BWC. They were making offensive bioweapons.
This is not Dr. Smith, making a small error and having an anthrax case. This was an illegal biological manufacturing facility that spewed anthrax into the air, killed a bunch of their own people, and then they tried to cover it up. Yes. And that is a very different, that is not a lab leak. That is like the opposite of a lab leak.
But
very
can I also just because there is the really key thing here, right? Which is that nobody is arguing that lab leaks don't happen, right? It's just, if we look at how often do lab leaks happen versus how often do random viruses spill over into humans, it's I don't even know how many orders of magnitude, but it's literally a billion to one, right? So that's the first part, is that because Matt makes the point of saying it would have been the first time that somebody would have been infected by a Sabeco virus from a bat. That's not correct, right? We know in the kind of villages in which you have these kinds of bats and viruses, is that lab leaks, zoonoses. Yeah, see how surely now I'm really covering something up about the labs out in the villages. No, is that these kinds of spillovers happen, frequently and on a daily basis, right? So we know that happens all the time. It's just they don't get the chance to take off, because many of the viruses don't have the ability to do so. But even the ones that may have the ability to do so cannot do it because you are in a very rural area in which the human density just isn't high enough to cause onward transmission. And that is why you need a place like Wuhan, right, which is an extremely dense urban center. Now you do take those animals, and now you don't just take them from the villages, you put them together as part of the trade of wildlife, in which all sorts of different animals are housed together in terrible conditions that make them stressed. Their immune system's a lot lower as a result. They, as a result of this, get infected with all sorts of potentially human pathogens, right? Zoonotic viruses, and then take them into wet markets and sell them there, in which you have these dense clusters of the animals and the people. And if the people get infected from there, then the virus can take off, right? Again, in most cases, even that doesn't happen because, again, the viruses don't have that opportunity. But the last part about this that is also really important is that as we're discussing lab leaks of the past, is that it is important to understand that these are of known human pathogens, right? Things like SARS-CoV-1 did not cause lab leaks prior to SARS-CoV-1 causing human cases. Once it had caused cases, those viruses get isolated, grown to high titers in labs, and as part of those, sometimes, yes, lab leaks have happened, right? But SARS-CoV-2 is completely different because it's a novel virus. It's a virus we had never seen prior to the pandemic. And now you are saying that a random bat virus that was taken to a lab then caused infections in individuals that then went on to cause a cluster over at a wet market 20 kilometers away. It's that part where we're saying it's not just a simple lab leak, and the comparison to prior lab leaks that we know of is basically off the table, because we're not talking about whether a lab leak could happen. We're talking about whether SARS-CoV-2 and the emergence in the human population could have been a lab leak, right? And these are not the same, and we should not confuse the two.
No, and we definitely should not conflate what people who are actually in the profession, we don't use the term lab leak. I think, I don't remember if it was you, Gigi, that I talked to about this like a few years ago, that prior to 2020, nobody used this term at all.
And we came up with it.
Yeah, I mean,
I was really wondering that. I would love to do an analysis of that, because I think it's really suspicious that it kind of came as a term.
Research associated infection,
Yeah, I mean, we call it a lab acquired infection, or research acquired infection, depending on the circumstances in a lab. That's all I think about when I go into a lab, is how to avoid a lab acquired infection for myself or anybody else, even more than a containment breach, actually, because it's pretty hard to unintentionally remove a pathogen from a high containment lab. But it's easy to remove it unintentionally if you get infected with it, because you probably don't know that you're infected with it.
Right.
So when we work in labs with dangerous pathogens, whether we know that much about them or not, we always take these precautions to make sure that we're not going to get exposed in there. And as I mentioned earlier, we have medical surveillance programs and all sorts of redundant systems to catch that if it happens very quickly, which, for example, the situation he brought up in Taiwan with both SARS-1 and SARS-2, those were caught because people were working in labs and they reported their symptoms. And that's why we know about them. I don't think that we do have, like Matt said, a bunch of COVID infections from working in labs, because most people still actually work with COVID, with SARS-2 at BSL-3, even though like you don't have to anymore. And I have to say, I've gotten COVID once. When I got COVID, I did report my symptoms, but I've been traveling for a week prior to getting COVID. And I know that I did not get it in the lab. And there was no debate at any point as to whether I got it in the lab, because it was like literally impossible for me to have gotten in the lab. But we still go through the procedure to make sure that I didn't get it in the lab, because that is responsible biosafety. And they talk about this like, what? We're just going into this little room over here, and I'm just going to do what the easiest thing to do is. I'm not going to really do that much paperwork or reporting. I'm just going to cook up some stuff and see what happens and hope I don't get infected. Like, there is no way that is the paradigm. And even if they didn't have as much experience at the Wuhan Institute of Virology, there's no way that they were doing stuff like that either. One of these State Department memos was actually the Wuhan Institute of Virology asking their US and French collaborators for help with more additional training, because they weren't certain that their biosafety procedures in their BSL-4 lab, which is not where they were doing the coronavirus research, except in non-human primates, they were worried about working with Ebola and things like that. So they were asking for help, actually. That to me suggests that they were very safety minded, and that we weren't even thinking about this like accidental lab leak kind of thing. Because
no, but the thing, Angie, I mean, a really key point there, right, is that like this idea that China somehow does not know how to handle these kinds of viruses, and somehow we do, right, is that the Wuhan Institute of Virology is a BSL-4 lab.
Yeah.
So they care a lot about biosafety in general, right? And they're extremely competent scientists. In fact, the labs we're talking about here, they're the ones that figured out everything around SARS-1, right? Which taught us about sarbecoviruses and the risk posed by these kinds of viruses, right? Is that these are not some backwater scientists that are just, bouncing around in a lab, right? These are professional scientists like many other scientists. And of course they know what they're doing, right? And to be clear, a lot of the work was done in BSL-2. And as you mentioned, that is actually the approved biosafety level for tissue culture work. But the animal work, for example, was done at BSL-3, right? Which is why you might get even higher titers. You could have respiratory, transmissions as part of that, was done in BSL-3, right? And this idea that somehow this was at, desperately unsafe conditions and it was done by people that have no idea what they're doing is just not correct, right? Like, these are very competent scientists, and the Wuhan Institute of Virology is a very competent virology institute.
And I think it was really telling where they come down on this when Matt Ridley was talking about how Zheng Liixia like responded to thinking that it might be a lab leak. Yeah. And he said, she started taking serology samples from everybody and looking through her lab and trying to see if she had anything like SARS-2 in her collection, and she didn't. Then he's like, well, that's very good and like responsible of her, but how can we believe her? And it's like, it always comes back to, but Chinese. Like,
yeah, but imagine that she hadn't considered the possibility, right? And I will say, like, this is something I actually brought up with my proximal origin authors too, where when I saw that article about Zheng Li, right, I was like, oh, that's she herself believed it was lab. And I remember Eddie Holmes educating me, and it was just like, well, of course she did, Christian. Imagine if she hadn't. And I was like, oh crap, you're right. Yeah. Of course she did. Right. And it was me for a second there. It was just like not functioning properly up here. Right. And Eddie rightly points out that of course she had considered that. Right. Imagine you had the same article and she would have said, it couldn't possibly have come from my lab. So I wouldn't even ever have considered it. Right. That would actually be a general problem. But that's not what she did. Right. She considered that possibility herself, and I'm sure took it very seriously.
So if we have a leak of 1918 out of NIH, I mean, I wonder if we'll get a response from Bhattacharya. There's no way that could have happened here. Must be a terrorist or something, because there's just no way. And I feel like this would be the response from this group. It'd be, that couldn't happen. We were silenced. This is, just making stuff up, basically, because there is no response to it.
Right.
Well, one thing that they bring up is like, they just want more information. Why are you not being transparent enough? And they first of all, like you said, Christian, you can't prove a negative, right? If there's nothing there, there's nothing there. There's nothing that you could show that would show more that nothing was there, right? But one of the things that they say is like, well, why don't you share the lab books? Why don't you share this? And I think it's remarkable how much has been shared, because if you flip it, and if you think about what if this was a U.S. lab, especially a U.S. government lab, like this would just never, you would get a certain amount of transparency, but you would not ever get lab notebooks and that sort of thing. And the reason I know this is because I was part of an investigation into when Dugway Proving Grounds accidentally sent out live anthrax all around the world. In 2014. Whoops. And it was, it was not intentional. Nobody was harmed. It was, they just didn't irradiate the cultures and for a sufficient amount of time. But like, we went there and they made a big deal saying like, see how transparent we are. We're having this woman who is from, at that time, University of Pittsburgh Medical Center, come and be part of this. Never mind the fact that like I had a top secret security clearance. I was part of a DoD advisory committee. Like, so I was not an outsider. You know what I mean? Like, so they would never ever let a Chinese national be part of that delegation. They would never let any other national be part of that delegation. And so to demand that of the Chinese is really not reasonable.
No, I think it's a double standard, right? Which we wouldn't ourselves be able to follow. And I'll also say, like, would I prefer to see all the lab notebooks of the Wuhan Institute of Virology? Of course I would. Like, I would love to see those things. In a perfect world, there was a lot of things that I would like to see that I don't currently have access to. But there's a difference between, like, look, here's the long list of things we would possibly want to see. But then taking it and saying like, but because we haven't seen it, we can't conclude something whatsoever. Hence, it must have come from the lab, right? That's where it's completely breaks. And especially as we are talking about evidence, there's a lot of calls for like, we need to see all the data from November. We need to see all this. Like, but look, there isn't any evidence from November, right? Not because they're hiding it, but because that there wasn't an outbreak at that time in period in time, right? And you're never going to get that evidence for the simple reason that it never could exist. But they have then done saying like, well, could there have been lots of infections in November that we could have missed? they obviously asked that question. They're like, well, we can address that by looking at blood bank samples, something that was suggested in the first WHO report. And the Chinese scientists did go back and investigated that to see, like, could we see any evidence of prior infections with SARS-CoV-2 prior to the detection of the outbreak in December? And the answer is they didn't see that, right? In fact, they only start seeing seropositivity in January of 2020, which means that it probably started in late November, early December, which is how we estimated based on genomics, cases, hospitalizations, and everything else, is probably the time at which this started, right? So again, these grandstanding demands of saying, like, unless we get all of this, we can't conclude anything, right, is really unfortunate, because of course you're never going to be able to get that information.
Yeah, and I think, I mean, they fundamentally actually don't understand phylogenetics, and they misunderstand it worse than I do. You know, I've read the Pekar paper like probably 15 times at this point, and I still can't really explain it very well, but I think I understand it. You know, this stuff is really challenging, and it's really nuanced. I mean, the same way that pathogenesis is, and I'm sure that all of the phylogenetic, like zoonotic, would probably have a very difficult time discussing like ins and outs of mechanisms of pathogenesis with me. Right. Totally fine. But it's a really complex area. And I just don't think, you know, I wouldn't be like, well, I don't know about that Christian Anderson's calculation of the mutation rate. I think that I'm going to go with something a little different, because you know what? He hasn't seen all of the November data that might not even exist. And how do I know that he's actually calculated this correctly? Right. You know, you said it earlier, Christian. I mean, this is a fundamentally unscientific premise, because you cannot prove a negative. And that's essentially what all these additional demands for data are. They're saying, like, prove that this doesn't exist, and then maybe we'll believe that it came from the market, rather than looking at the fact that the entire evidence base shows is consistent with a market origin and not with a lab origin. There's only one piece of evidence that suggests that it could have been from a lab, and that is the fact that the pandemic started in Wuhan and that there is a lab there.
Correct.
That is it. Yep. There is nothing else that is consistent with a lab origin. And the fact is, we have been saying this over and over again for the last 6 years, saying it progressively with more detail as we have gotten more detail, as we've continued to do more analysis. And we actually feel that, you know, it's pretty strong, but it's hard to make a scientific argument against this when they say, okay, gold standard science means actually doing completely unscientific things like proving negatives. And also, we just don't trust you very much. There's been a loss of public trust. Didn't you know? All of you are untrustworthy. You naturally, as virologists, as human beings, want to continue doing the work that you're doing, and you just love doing it so much that you don't want us putting any restrictions on you or saying no more virology or whatever, which is also what they think our first instinct is. I mean, that was not my first instinct when I was like, could it have come from a lab? I was like, how would we determine that? Like, how would you investigate?
Even if it did come from a lab, even if it did, if the evidence pointed that way, I'm still struggling to understand how that has any effect on the biosafety precautions taken anywhere else in the world. You know what I mean? And so this is like, it is to fit the narrative that Trump decided right from the very beginning that it came from a laboratory, and then people fell into that line, and for various purposes that they have to have it come from there. But, you know, it should not necessarily be the case that just because the Army sent out a bunch of anthrax without irradiating it doesn't mean that, you know, that other labs wouldn't have taken more precautions.
And, you know, Gigi, I think what you just said about Trump decided, I'm not sure that that's what happened, but I do think that what happened, and this was something that became very, very clear to me when I saw what happened to the COVID.gov website. So, you know, the COVID.gov website used to be a website where you could get information about finding COVID tests. And now it is this insane website that says lab leak in the craziest fonts on top of it. And like Trump is striding out like through it, like looking like some sort of, you know, like juiced up, like propaganda god, except it's Donald Trump. And then it has the whole basically COVID-19 select subcommittee report beneath it, which is like lab leak. And then also we hate Governor Cuomo. And so that part doesn't really make sense for the lab leak. But as soon as I saw that webpage, I was like, okay, I see where this is going. Because what Trump did decide was that he was on board with Project 2025 and with Russell Bott's attempt to basically cut as much of the federal budget that is congressionally appropriated as possible, illegally impound it, and redistribute it to the executive branch, thereby centralizing Donald Trump's power and eroding American democracy further. And that was when I realized that that is how the lab leak is going to be used, because it is being presented as this was Biden's fault. These are the Democrats doing this, and this is what caused the pandemic. Never mind that, of course, Trump was president when the pandemic started, but whatever. It was somehow Biden and Obama's fault anyways. The real,
you know what I thought when I saw that website? I thought, great web design. I was like, that looks really good.
I loved the calligraphy font. It looked like a wedding.
I think it looked really good.
It was
the calligraphy shows to me that it was done by somebody over 40, because I can tell you that my kids can't read cursive. So that's,
Well, I just loved it. It was like lab leak. And then it's like part XO Donald Trump.
But Angie, can I just let me just think, because you touched on this in the beginning, right? Where again, the scientific inquiry where we're talking about here, which is that, like, when I talk today about a lab leak and I talked about zoonosis, right? I am very confident in my beliefs on what I believe happened here. But the really key point about that is that it's not because I have done all the analyses myself and therefore they must be true and we should always trust what I say. I'm not the lone ranger here, right? That sort of strode the path through what this happened, right? The reason why I am confident in our conclusions is because I know that I have worked with a large group, an ever larger group of other experts that have different kinds of expertise than I have, right? So when I have the key things that I can look at, but then it's like, well, and I think over here, but I'm not really sure. We are always working with, several people, not just one person, right? Several people who are bona fide experts in exactly those kinds of questions. And then we collaborate together and publish papers together. And that is why, like, when somebody like me, Matt said, all the scientists completely failed and it was only the amateurs that could do the right research, right? Is that when you hear that, you got to ask yourself the question, which is, really, right? Because do you really think that as we're looking at these kinds of analyses, which are quite technical in nature, right? Very technical in nature, is that, do you really want random, internet sleuths be the ones that do it? Or do you want scientists collaborate together with each of their own expertise to go towards their conclusions that are importantly then published and peer reviewed and can then be critiqued from there? I think this is a really big difference, which, is important to understand about the whole, do your own research. I completely agree, but you can't do all the research on your own, right? You need to do that with other experts.
Yeah. And
I'm sure.
Oh, go ahead, Gigi.
I'm sure that Matt Ridley, if he were to have a health event that required the kind of scientific expertise that to figure out, he would want to have just that. But there are people though that's not necessarily the case, and they would rather go with like the hunches and to figure things out, because the rhetoric, and the argument that you can make with words is to them more valuable than the scientific evidence.
Yeah, and if you look at like how he started his talk, right? Because the first thing he does is to discredit the evidence, right? And saying like, you can't trust any of the evidence. Yeah. It's all biased. So that then takes care of all the published papers, right? Peer-reviewed journals, saying like, okay, can't trust any of that. It's all biased. Trust me.
Science and self.
Except of course it's not. And then once he has done that, the next thing he then does is they're saying like, and now let's talk about the scientists themselves. Yeah, you can't actually also trust them because they published all these shoddy studies. And of course they would do that because otherwise they would lose their grant funding, right? That's then step 2. And now you have fully discredited any level of expertise, any level of expert, any level of scientists that might have actually looked at this with critical eyes, right? Mm-hmm. They have now been dismissed. And now it's all game on, right?
Because vibes only.
Yeah. And now you get what you get instead are these scientists like Matt Ridley and like Jay, who have doctoral degrees and who appear, you know, Matt got his doctorate at Oxford. Jay was at Stanford for many years, which is also where he got his degrees. I mean, they have credentials. And the average person, you know, looking at the four of us is probably like, they all work on the same thing. And I mean, you know, Christian and I are both biologists, and like we both have a background in immunology, and like we do work on the same thing, but in a completely different yet related and complementary way. We just work on different parts of, you know, the entire complicated mess of viruses and spillover and all of that stuff. And so one of the things that I've experienced, like working with the Zoonotti, is that it has actually been, I mean, one of the most stressful things during, at least during my pre-Trump post-pandemic experience, but also like one of the most rewarding situations. Like I actually have learned so much about phylogenetics from Christian and Mike Warby and Andrew and Eddie. I've learned so much about virus evolution. I like to think that I've helped people understand better how laboratory virology work is done. And, you know, we've all had our own contributions to make. We all have our own perspective. And I think that has really made the science better. I don't think any single one of us could have done this on our own. And importantly, none of us could have done this by like Googling for shit, you know, and writing blog entries about it.
But you could do what they did for just Googling stuff, you know? And
I mean, I could find some theses and toss them into a translator and, you know, find out what one of Zheng Li's master's students was doing 15, 20 years ago. But I don't really think that's relevant to the question, because I'm not, you know, I do that. That's my research project when I write a Substack blog. Okay. Like, yeah. That is not my research like method when I am actually doing a scientific study, because even if I'm doing something that's strictly computational, I'm still framing it as testing a hypothesis, which is how the scientific method works. The scientific method is not trying to prove yourself right. It's trying to prove yourself wrong. And so like people really don't understand that when Christian changed his mind, it's because he was actually doing science the right way and he disproved his initial hypothesis, or it failed testing. And like that is how normal scientists do things. We don't find an amazing guy in India named the Seeker, which, you know, obviously is not his government name, who's been tweeting a lot of cool stuff. And so therefore he knows more than Christian Anderson about virus evolution. I mean, this is the dumbest logic in the universe, but it appeals to a political audience rather than a scientific one.
It also appeals to Jay Bhattacharya, who kind of went into, you know, he rose to prominence by coming up with a conclusion for how prevalent SARS-CoV-2 was, and buttressing the numbers to get the result that he wanted in his Santa Clara study. So it's like, you know, that's not the way you do science either.
No.
I think this is just really devastating in that I think it's really like to the end, like this is something that the NIH, like this is not just like a random presentation that we're seeing. This is something that the National Institutes of Health, the largest biomedical research funder in the world, is putting on and saying is valid and is like really worth listening to and is scientific. And like I think this panel that we're doing, I mean, y'all are really answering really good questions. I think it's so important because like that, just what this is just a breakdown of the public's trust, both in the NIH for people who are maybe more like scientists, the NIH and what it is currently, and then for the public, the NIH and what it used to be, and then only trusting what is coming out of the NIH and really what's coming out of Jay Bhattacharya's mouth, because he's saying that no scientists who worked here before this are worth trusting. And so when people are speaking out about the changes that are happening, it's like, oh, well, Jay's saying that they're not worth trusting because like I have to do the second scientific revolution. So like nobody's worth trusting other than these people. And I just think it's really devastating because it's like distracting from some of these really horrific changes that are happening. Both because now everybody's like, oh, well, there was this disinformation talk. And then also it's just like putting the public's attention on this talk as opposed to everything else.
Yeah, I think it's a really key point, right? Because like this is supposed to be like, the scientific freedom type of lecture series. And this is now setting like, and when we're talking about how science should be done, then what is being pointed at here is Matt Ridley's lecture, right?
Yeah.
And it's a terrible precedent, right? Because like, again, none of it was scientific, right? To be very clear, right? There was no science in that talk whatsoever. Matt himself has not done any research into the origin of the pandemic, right? He's written a book on it. He has not published any peer-reviewed journals, which he puts down to it's because they're trying to silence me, right? Is that it sets a terrible precedent as we're looking to the next generation of scientists looking to be, what is science supposed to be like? And this apparently is gold standard science, right? And it is, right? It is instead platforming pseudoscience. And I think that is really devastating to see. And again, more than anything, I'm just extremely sad to see it. Right.
Yeah. I see this as, so I was raised in the US. I think Christian and I are the same age. We were born the same year. So I'm like, middle-aged. And I grew up in the 80s. Yeah, you're not. Because you're from Denmark. So like you guys never age. You just
25 forever.
You just stay perennially inferior to Norwegians.
Do you really want to be 25 forever though?
We can have a whole other conversation about longevity another time. But oh man, now I'm like, I forgot what I was gonna say. But I think that what I wanted to hear actually about earlier, Anna, is how this makes you feel, because as somebody who worked at NIH, you and your colleagues there, who are not just like intramural scientists, but are actually running these programs. I know how hard you work. Everybody else who has worked with NIH program people know how hard you work, that you guys are great scientists, and that on top of that, you really care about the impact of the science that you fund. I have never had an experience with an NIH program person who was not fully committed to the research that they were funding and cared and wanted me to be successful doing it, but also wanted, the results of that research to go on to feed more research. And as we all know, it has many benefits for the US economy, all of this stuff. And so to have the NIH director platform somebody like Matt Ridley, who in this completely unscientific talk, completely misrepresenting your colleagues, everybody throughout the NIH, all of the researchers that are funded by NIH money, in the service of essentially destroying the NIH. I mean, that has to be, I think all the time about how painful this is for me and how sad it makes me as an American, as well as a scientist, because, and my history, we don't have to go into long history, but I've had a real problem with shitty men through my entire career. And I feel like I continue to overcome these shitty men, but now I have like the ultimate shitty men to deal with. And they're going to take all of science out because of their shitty man ways, and also their complete unscientific devotion to a political system that is going to undo everything that is at the core of my values as an American. And I really want people to understand more than anything else, because I think you and your colleagues at NIH probably understand this better than anybody else, what the consequences of this will be. But most Americans, I don't think, understand that the NIH is not just like, give me and Christian and Gigi funding so that we can write papers and have a job and have a lab. Like, that is not the NIH's only function. And many NIH grants have huge clinical components. They are essential for building out infrastructure. It is a cornerstone of our economy, and it's basically responsible for every single new drug and vaccine that we have. So I don't think that people are truly clocking that this gold standard science bullshit is actually going to tank our economy, kill a bunch of people, make it so that a bunch of hospitals close, and make it so that we don't have any new doctors or scientists to do any of this work in the far future. And that's why you can't just restore a grant or say, oh, we're not cutting the whole NIH budget, when there's all these other things, and we haven't even gotten into forward funding. That's another conversation.
Yeah.
But when you have the NIH director spreading disinformation, lying to the American public about science that his own agency has funded in the past because he has a political disagreement with the previous ownership or the previous management, I mean, that is not only unscientific, in my opinion, because of what the consequences are, that is criminal. And my goal for all of these things is to hold these people accountable, because one day there will be a reckoning. And you know what? We are staying true to our training as scientists, and we're at least collecting the evidence. It will be there when the time comes to review it.
Yeah, I think that just to talk, so I was in peer review. I wasn't in program, but I know a lot of program officers, some current and former program officers, and people who work in a bunch of different places at NIH, intramural, extramural, and the Office of Director. And I think that across the board, it's just really sad. It's super demoralizing. It's terrifying. I mean, we're seeing, how many people are going to die because of this, because of the stoppage and interruptions of clinical trials. I mean, and every single day it feels like we're responding to a different crisis, because maybe it's impacts on animal research, it's impacts on international collaborations, which again, also this is such a further reinforcement of the xenophobia of this administration. And it's going to kill science. I mean, we need international collaboration for so many different reasons, especially when you think about rare diseases. If there's only a patient population of a particular number in the US, you need international collaboration to be able to look at those rare diseases. But it's just really, really hard to listen to, because, and I think it's hard too, because I think if I turn off my brain as a person who worked at the National Institutes of Health or has a scientific backing, I can listen to some of the things, and not necessarily this talk, but other things that I've heard Dr. Bhattacharya say and other leaders from the NIH, and just it can sound appealing in some ways if I just turn off those two parts of my brain, and it's it feels like I'm just in a completely different universe. I feel like I'm just in such a different reality. So it's it feels like I can forecast the death of so many people, the premature death of so many people, because of the trials that are being interrupted, because of the research that's not going to happen, and because of the politicization and, someone's political agenda being thrown into all of the research. I mean, the DEIA, getting rid of DEIA, it impacts every single part of the academic industry. And I think we're watching academia change fundamentally for the worse. And it's already done decades worth of damage that it's going to take so long to be able to repair, both as scientists and our trust with the public, because every single thing that comes out of Dr. Bhattacharya's mouth publicly is further making our job. It adds on, I don't know how many days that we're going to need to rebuild that trust with the public, but it's just such a decimation.
Because he's lying nonstop. I mean, it will decrease trust if you constantly lie to people.
And you say that you're not lying. So you're I'm the only one who's not lying, but you're lying while you're saying that you're not lying. And you're throwing these grains of truth in the things that you're lying and not lying about. So it's devastating. I mean, it's horrible. Everybody I know is at the breaking point of how are we going to keep doing this? But we keep fighting back and speaking out. And there's so much power in what we can do if we continue to be honest and transparent about how awful these changes are, because we don't see everyone doing that. Not all organizations or scientific leaders are speaking out about how horrible this is. And it's important that we call out when Dr. Bhattacharya lies. I think that the people who asked the questions today were super brave. The people who were willing to push back, because as a person who worked in the government, that would make me think I was going to lose my job. I would be afraid if I was them that maybe I'd lose my job, but it was so important that they did that. So it's a long response to how it makes me feel, because it feels so awful.
I think it's really important though to hear responses like that, because I don't think that people understand the human cost of this. I have been going through a very difficult time, partly because of the lab leak, but partly because of everything. I live in another country now, and it still affects me profoundly. It has had a profound effect on my physical health. Gigi can actually vouch for this, because she saw me when I was about to have a serious medical problem last week that I didn't know about, or 2 weeks ago. And it has really impacted all of us. And I don't think people hear those side of the story. They never hear that side of the story. They see Christian testifying in front of Congress, and then they talk about how evil Christian seems. And don't think about Christian as a human being with a family who is profoundly affected by being dragged in front of Congress and turned into a spectacle so that people can further their own political objectives. And, you know, I don't think that people really understand what a toll this is taking on Americans who've dedicated their lives to either public service, not in my case, but in my case, I've dedicated my life to doing science, to trying to make the world a safer and healthier place. And I feel like everybody, every one of my colleagues, at least the people that I hang out with, went into it for the same reason. You know, they wanted to make a difference. They wanted people to be healthier. They wanted to know more about the world that we live in and make the world a better place. And that is what is being trashed right now by the NIH director. And it's just absolutely crazy. But I really want people to understand that this is not just a difference of opinion. This is not like people with an alternative scientific point of view are in charge. Correct. These people are political operatives, and they are deliberately trying to destroy the NIH, and they are deliberately trying to destroy American biomedical research, and they are deliberately trying to destroy American democracy.
And it's something just like, it's not a popular thing. When we see like the tangible results, like what happened to Vinay Prasad, I think was really directly a result of him stepping on what has long been like a political third rail. You don't trash patients who have rare diseases. Like you just don't do that. And when you have Huntington's patients who are there able to talk about their experiences, why they want to have that drug that has shown great benefit, it moves hearts, even people, and in a very bipartisan way. But the problem is with what happened at NIH is that it's a little bit too far upstream, and people know intellectually, or congressmen know intellectually, that it's important for the economy or that it's important for drugs, but you don't have that person who's going in front of them and saying like, I'm going to die soon because I did not have this intervention. And so I think, that's really why it's so important that we keep doing these kinds of things and telling people and showing people what science is all about, why it's important, because without these stories, what do you have?
I will say that the clinical center does do clinical, it's the largest clinical research hospital, I think, in the world. It's completely dedicated to clinical research. And it does, people were impacted. Yes. Like the clinical center, the entire NIH is impacted. And that in clinical trials, people have come forward, some patients have come forward and said, my clinical trial was impacted. I wasn't able to get my medication and my disease was impacted, all of these things, as well as there is a patient who passed away. There are things that are happening. It's just really hard right now because everyone's so scared to speak out. So I think, yes, with the FDA, it's just harder to, I think that getting patients and people to speak out about the impacts is much harder. And you're making a really good point about that. So it's trying to really amplify the voices of the people who do speak out and try to make the tangible connection between, this really important research that we're doing all the way to, how does this impact the American public?
Well, it's just a very important part that what you're doing, because, patient groups like the Huntington's groups, they've been around for a long time advocating for research funding. And, this has been a long-standing, people have known what Huntington is for a long time. And so these groups have been around to advocate, and that's what science needs more of, is advocates.
Yeah. And I was just going to say,
thank you.
We've been on here for over 4 hours and you guys are champions. And I don't have any plans immediately, so I could stay on, but I'm wondering if perhaps we might sign off now and think about doing this again, even if there's not another terrible lecture. And maybe
I have some takeout to pick up.
Angie, can we just for a few minutes here though, just to finishing up? I think I have a slide deck that sits there because I just wanted to again about Matt Ridley's talk today. I do think it's important that there's a few points that in terms of his conclusion slide.
I'm going to give you the floor and I'm going to give you the control of the slides. Which one do you want first?
The one that's called extra.
Oh, extra?
Extra, yeah.
It's called slide 1 here, but
okay. There we go.
Yeah.
Because I just wanted to just spend a few minutes here, because this was Matt Ridley's conclusions, right? Again, he says that in 2017-18, Viv shifted their priority to 20% divergent viruses. He used that as an argument for why they would be studying SARS-CoV-2 and not SARS-CoV-1. That is not actually correct. At the time, and he did this, in response to talking about diffuse. What is really important here is that he's mixing up two completely separate grants. This whole idea that they'd shifted their focus to 20% divergent viruses is not correct. The focus was still on SARS-1 and SARS-1-like viruses. And it's because he mixes this R1 from 2014, where they talk about, remember he mentioned this 10%, 25%, is from a completely unrelated grant to diffuse. Here's diffuse and here's that other grant. Matt just mixed the two and saying, oh, they've shifted all the focus to what would be SARS-CoV-2. It's just incorrect, right? He says they sequenced at least one lineage 4 sabecovirus. All those genomes are available, right? Again, they have been published, so there's nothing there. He said they proposed to insert a furin cleavage site into a sabecovirus. He specifically said that was S1, S2. Actually, if you read the proposal itself, as people like myself have pointed out, is that they're talking about the S2 cleavage site, right? Which is downstream of that. So again, the fact that we have pointed out that they're talking about that is just incorrect on Matt's side. He talks about proposing infecting human cells and humanized viruses with such viruses. No, this is basic virology research, right? There's nothing about that. At a low biosafety level, we have already talked about that, right? This is the generally approved biosafety levels. A lineage 4 sabecovirus appeared with a newly acquired furin cleavage site. Again, he is now mixing these two. Again, it's not the lineage 4 and the viruses they're working on, and nothing to do with SARS-CoV-2 in the very same city. Correct. This has been true since the very beginning, right? Which is why we need to consider a lab leak in the first place. Leaving no trace in any animal anywhere. Well, they haven't looked, right? For one specific thing is that they didn't actually look at the relevant samples inside that market. But really importantly, we do find samples from within the market itself in which we find evidence of the virus as well as susceptible animals. Viv misled the world about the name and the date of sequencing of viruses. Just outright false, right? They didn't actually. Just because Matt can't tolerate that they changed what was a sample name to the name of a virus, now they had the full genome, does not mean that they were trying to mislead anybody, right? This is the Matt just making it up. And then took the database offline. Again, he says that happened in September. It's not actually correct. Again, the database is live all the way up until early 2020. But also now he's proposing a September date for some sort of lab leak, right? But at the same time, he talks about how the first cases of COVID must have been in November and at the Wuhan Institute of Virology. Again, those two are completely mutually exclusive, right? You cannot have both. So I just wanted to, as we are looking at these things in which he concludes, and this is why he says that this is a lab leak, out of all of these, only in the very same city is correct, right? Everything else is basically a misrepresentation of the science and evidence itself. So I just wanted to leave that up there because I do think it's important as we talk about the conclusion, right, is that there's just no hold to any of what is actually written right there.
Yeah, that was amazing, Christian. And I'm really glad that you broke it down that granularly, but without getting too technical, which is a very challenging thing.
I could do that, but yeah, no.
Because I've also been reading in a separate Zoonotti channel, like your questions to one of our colleagues, to get all of this stuff together to just fact check. Yeah. And it's
actually one thing. The last thing I just wanted to, because he mentioned this ascertainment bias, right?
Yeah.
And he says that it's because it was required that cases had to have a connection to the Huanan seafood market. And I mentioned this when he mentioned it, but I just wanted to show the actual evidence. And this is from the WHO report, right? And you can see this issue date of the first case definitions in the mid-January 2020, right? Is that you can see under number 2, there is the direct and indirect exposure to related markets in Wuhan, right? But again, this is mid-January, yeah? And importantly, it also changes again on the 18th, where that requirement is removed. So just within 3 days, that requirement is removed. But what is really important here is that as we are talking about our papers and this clustering around the market, we're talking about December cases. right? We're not talking about January cases. And of course, all of those are retrospective investigation as part of the first WHO mission to get those December cases, because of course, at the time, they didn't know they had an outbreak. So the idea that this is all ascertainment bias caused by these definitions is just not correct. That definition was there in mid-January, and it was only there for 3 days. And it is obviously irrelevant as we're talking about the cases in December of 2019.
I couldn't agree more with all of this. And I'm so glad that we were actually able to really respond to stuff like this today, because it is very difficult, as Anna, as you can probably see, and as everybody watching who hasn't been following this, there are so many parts to the story, and this stuff has been going on for 6 years. And not only is the science itself incredibly complicated, but dealing with all of the lab leak people and all of the stuff that they've been putting us through, especially Christian, has also been really, really challenging, because it's essentially been like playing whack-a-mole with our own data for the last 6 years, where somebody says, what about this? And then we say, not true. And then it just turns into, but yeah, what about this? Other thing, as Christian pointed out earlier too, many of these different hypotheses they sort of ping pong back and forth from are not even consistent with each other.
They don't have to be.
It matters.
No, they don't have,
it's
right,
not
they don't have to
actually
be.
scientific. It's a simulacrum of a scientific process, but it's pseudoscience. And the reason for it, again, I think, is that they are going to use this as the excuse to completely get rid of most of the NIH's research budget. And very depressing way to end.
Well, a better way to end is that it's good that we did this, you know, and we have to like keep pointing this out.
Yeah.
The reason that we can do this is because we do have a coherent story, and they are using pieces of it when they choose to, showing your papers and how they're interpreting the data, because they can't.
And yours, Gigi, about Sverdlovsk.
I want to, just so that we're not ending on such a like really scary note, right? I just do want to say that I think that, you know, things like this, everyone's speaking out as much as they possibly can, I think is really important. And there are so many people who are like behind the truth. And so I think the more that, because we're in such a culture of fear, I think both in academia and in like in the government, I think the more that we can say that there are people like me who in the government and people like you all in academia who are speaking out and being transparent about the things that are happening, that we can just like continue to encourage people to find community and like work with the people who they work with to continue to speak out. You know, the threats that Dr. Bhattacharya and these lectures bring to the public are going to continue to be diminished the more that we're able to speak and be honest about them.
I totally agree, Anna, and I couldn't agree with you more, actually. And that is the reason why I have been focusing so much more on communication, and why I was so grateful, actually, to Jeremy Berg and Stand Up for Science for approaching me about wanting to do something like this with me and the Save America movement and my colleagues, and then with you and your colleagues at 27 United. This community came together because there was really a need for this session. And I want to say, really encouragingly, it can be very difficult to be one of the only people speaking up, as I think the four of us probably know. But this has been going on for almost 4.5 hours, and we have actually been steadily increasing viewers. I've been watching. About an hour and a half ago, we had around 900 viewers, and now we have over 1,200. We have progressively been acquiring more and more viewers as this has gone on, which, considering this is a 4.5 hour meeting, that included a scientific lecture where we're talking about like very detailed aspects of origin science, I think that's actually pretty impressive. We just got another 5 viewers. Hi, guys. We're about to sign off.
Well, that's a lot more viewers than were in the audience for Matt Ridley's talk.
It sure is. And you know what? That gives me a lot of hope, and being part of a community like this and seeing how fast a community can come together when there's a need for something like this, seeing how, you know, people can give each other strength and courage. And when they see one person speak up, it makes it easier for other people to do so too. And that makes it easier for everybody. So I'm really, really grateful to all three of you for joining me for this. I'm so grateful to the Save America movement, Stand Up for Science, and 27 United for really helping to put this thing together. And I think we'll talk amongst ourselves and maybe make this a recurring series. Hopefully there won't be that many more of these horrible lectures, but if there are, we'll be here to debunk them. And if there aren't, I think that it's not a bad idea for us to get together regularly and have conversations like this, because we do need to be discussing this as an entire scientific community. We also need to be communicating this to the larger community of the public. So I'm just really grateful for everybody, and enjoy your weekend. Have a great Friday night, and it's 6:30. There might still be time, at least on the East Coast, there might still be time to make a happy hour somewhere.
Absolutely.
Thank you,
Angie.
Anna.
Thank you all so much.
Thank you guys so much. I'll see you soon. Take care,
Bye.
everyone.
Bye.